A very big hello to my wonderful PU2 champions. I hope you're doing absolutely great. So there's only one thing that I want to tell you before I start off this video, and that is no one can stop you from scoring full on full in biology, because my dear children, I am going to teach you, and I have full faith on myself that I will give my best. And I have full faith, more than me, I have faith on you, my dear children, that you are also going to give your best. Okay, so I hope you're ready. We are going to cover the six remaining chapters, that is Microbes and Human Welfare, and Biotechnology Principles and Processes, Biotechnology and its Application, Organisms and Population, Ecosystem and Biodiversity. The rest of the chapters, the initial chapters starting from sexual reproduction and flowering plants to human health and diseases, have been covered in the live session. So if at all you have missed it, watch the live, you will get all the important questions. Okay, and these hours that you're going to spend with me, I'm telling you, whatever marks these chapters are going to give, you will get the whole of it. That's my guarantee. Just have your notebook and a pen with you guys, that's all. Okay, starting off this particular video with the MCQ questions of the chapter Microbes and Human Welfare. You can expect 2 MCQs coming from this chapter. The first question from model question paper 1, and that is Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting malted what? So Saccharomyces cerevisiae, which is there, it is the yeast which we used for baking also. In that industry you call it as baker's yeast. Over here you are using it for brewing, so you will call it brewer's yeast. But what is it used for? Basically for fermenting malted what? Malted pulses, malted cereals, malted fruits, malted vegetables. It will be cereals. A very small line given in your NCERT. Okay, that is, yeast is used for fermenting malted cereals as well as fruit juices. What do you mean by malted? That means they have started off germination, but they have not completed the process. They have got stopped somewhere. Why do we need malted cereals for fermentation? Basically, when you start off germination, you break down the complex sugar to simpler sugars. Okay, so now the breakdown has been officially started. You don't need to put extra efforts. You get it? It makes easier to ferment malted ones. So malted cereals are being used. Always remember that. Okay? So the next question, cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogenous. So you're talking about a cyanobacteria which shows autotrophic mode of nutrition, as in it can prepare its own food. Now you have been asked the question, that is, there's a cyanobacteria, it is an autotroph, and basically it can fix atmospheric nitrogen. That means it can take the atmospheric nitrogen and make it into different forms such that the plants can easily use it. So what is that cyanobacteria? You have amoebiasis, richness, nostoc, ovulation, colostrum and diversity. Your answer is nostoc. Remember, you have 3 examples given in your NCERT. One is nostoc, the other one is oscillatoria. Okay, and the third one is Anabaena. So nostoc, Anabaena and oscillatoria, 3 of them are cyanobacteria, and 3 of them are responsible for fixing up the nitrogen up there. Okay, cool? Yes, and remember, the cyanobacteria is kept different from the other bacteria out there. There are other bacteria also given in the NCERT, such as Rhizobium, Azotobacter, okay, Azotobacter, Azospirillum. All of them are kept separately. Cyanobacteria are kept separately. Okay. Next, because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk? What is there in the curd that makes it nutritionally richer than milk? It is direct question. Well, the answer is vitamin B12, which is why you should also be having curd. You will get vitamin B12, my dear children. Who produces vitamin B12? So while the milk is being converted to curd, no, you need a guy. The guy's name is lactic acid bacteria, or example of that is lactobacillus. Okay, so while it converts, it also produces vitamin B12. Now it is there in the curd, you can just have it. Okay, so your answer for this question becomes what? D. Now, at any point, if at all you feel the speed is a bit high, make sure you go to the settings and make it a bit less, because over here, I know that you have time constraints, which is why I'm speeding up a bit. But if at all you have problem in understanding, please make sure you go use the settings. That's for you. Yes, because this video otherwise will be very long, and I'm pretty sure you do not prefer very long videos, right? So, yes. Okay. Mycorrhiza is involved in absorption of dash from the soil and passes it to the plant. You know, mycorrhiza, cyanobacteria, bacteria, all of them are biofertilizers. They help in increasing the nutritional quality of the soil and basically helping the plants with the growth. They act like biofertilizers, my dear children. So mycorrhiza over here is a combination of fungi as well as the root of the plant. What does this fungus do for the plant? What does this fungus do for the plant? Fungus basically gets into the soil and absorbs a very important thing, which is known as phosphorus, and gives to the plant. What does the plant give it back? Plant gives organic matter because plant is able to do photosynthesis. So always remember, mycorrhiza is involved in absorption of phosphorus from the soil and passes it to the plant. This type of relationship is known as mutualism, and this is a symbiotic relationship where both of them are helping each other. Okay, and also very, very important thing, apart from giving phosphorus, there are other functions also provided by fungi. Fungi provides resistance to pest. Okay, fungi provides tolerance to salinity. That means if too much amount of salt is there, still the plant will be able to survive. Fungus provides tolerance to drought. If there is no water, there is less water, still the plant will be able to survive with the help of fungi. Fungi also helps in overall growth and development. There are 4 things. Always remember the 4 things. Apart from that, it does not help in increasing the yield of the plant. Yield of the plant is not increased by fungi. This is a usual MCQ question, which is why I just discussed it. So your answer for this question is phosphorus. Remember, this is also an example of mutualism from other chapter, that is organisms and population. And this is a biofertilizer. Mycorrhiza is a biofertilizer. The fungi belongs to glomus. Okay, that's a species. Okay, next. During sewage treatment, the gas produced in the anaerobic sludge digester includes, if at all you're talking about biogas, always remember, biogas is just made up of 3 things. That is your methane, H2S, hydrogen sulfide, and also CO2. Now sometimes instead of H2S, hydrogen is given. Will the answer be wrong? No, it is correct only. So CH for methane, hydrogen sulfide or hydrogen or CO2 makes up what? The biogas. And always remember, biogas is produced only by anaerobic bacteria. Okay, anaerobic means the ones who do not need oxygen. So biogas production happens in the absence of oxygen by anaerobic bacteria, and these are the 3 things that you should not forget: CH4, H2S and CO2. Biogas is not just made up of methane, but those 3 gases. So your answer for this question, during sewage treatment, the gas produced in the anaerobic sludge digester includes what? Methane, hydrogen sulfide and carbon dioxide. So your answer for this question becomes C. Okay, it is C. Next question. Dash is a producer of butyric acid. What produces butyric acid? The closest name that we have over here is Clostridium butylicum. So yes, my dear children, it's a bacteria and it produces butyric acid. This is example of microbes in industrial usage. Okay, so Clostridium butylicum is a producer of butyric acid. Going ahead, so over here you have different names of other chemicals also. For example, citric acid is produced by Aspergillus niger. Acetic acid by Acetobacter aceti. Ma'am, do I need to remember names? Obviously yes, you would have to. We will cover them also in the 2-marker, 3-marker session, so I'll keep it for the end, or do you want me to discuss it over here? Let's discuss it over here. So acetic acid, Acetobacter, butyric acid, Clostridium butylicum, lactic acid, Lactobacillus or lactic acid bacteria, ethanol, okay, alcohol. I told you yeast is used in the production of fermentation where it produces alcohol as well as CO2, right? So Saccharomyces cerevisiae, yeast is being used. Over here, please remember 2 things. Only 2 places fungus is there. One is citric acid, which is produced by Aspergillus niger. It's a fungus. Over here, ethanol, Saccharomyces cerevisiae, yeast is also a fungus. Rest all of it, bacteria. Okay? Yes. Now, Propionibacterium shermani is used in the production of Propionibacterium shermani. It is shy. Shermani in Hindi. Sharmana in Hindi means getting shy. So Propionibacterium shermani is shy because it is releasing carbon dioxide. And when it releases carbon dioxide in the cheese, my dear children, in this cheese, it gives rise to what? Big pores. Big pores. So that is why in the cheese, which is known as Swiss cheese, in the Swiss cheese, you have big pores because there is release of carbon dioxide by this bacteria, that is what? Propionibacterium shermani. So it is not Roquefort cheese, it is not, okay, it is not bamboo shoots, it's not even ferment fish, it is what? Swiss cheese. Swiss cheese. Remember that. Okay, and Roquefort cheese is also produced by a fungi. It is produced with the help of fungi that is given in your NCERT. The name of the fungus is not given, but please know it's a fungus. Over here, it's a bacteria. Okay, S for Swiss cheese. You can remember like that. Yeah? Yes. Why have I marked this? This is the answer. Okay? Next, Alexander Fleming discovered penicillin while working on. Alexander Fleming discovered penicillin while working on what, my dear children? Staphylococcus, Streptococcus, E. coli, Salmonella. Alexander Fleming was basically working on this guy, that is Staphylococci. Okay, Staphylococci, not Streptococcus. Usually children get confused. Don't have that confusion. Okay, always remember Fleming has an L, Staphylococci also has an L. You can remember like that. So basically he was working on that. One day he forgot to wash the petri dish. The other day he thought that the bacteria will be growing everywhere in the petri dish and out of the petri dish. But that did not happen. Why? Because a fungus mold was growing up there. The name of this fungus is Penicillium notatum. Okay, Penicillium notatum. And what is it producing? It is producing penicillin. It is producing penicillin. Always remember. So Alexander Fleming discovered penicillin while working on Staphylococci, Streptococcus, E. coli, Salmonella. The answer for this question is what? Sorry, the answer for this question is Staphylococci. And what was the name of the antibiotic being produced? Penicillin. And what is the name of the organism? Penicillium notatum. And it's a fungi. Remember, 3 people got Nobel Prize for this. One is Alexander Fleming, second one is Ernst Chain, and the third one is Howard Florey. So basically 3 of them got their Nobel Prize. And also this important antibiotic which was there, it was used in World War II. But who discovered it? Only one guy. Only one guy discovered it. Later, the other people got to have more understanding of it. That's all. Okay. Okay, this was Alexander Fleming. Now I have written it over here. You can go and read it. The PDF you will get. But for that, you have to join the WhatsApp channel. And if at all you have not subscribed to our channel, by the way, subscribe to it right now. So which one of the following alcoholic drinks is produced without distillation? Without distillation. So what do you mean by with distillation? Distillation means you are making sure the water is evaporated. Suppose you have a flask of something and you are making sure whatever water content is there is evaporated. Now whatever is left behind will be concentrated, my dear children, because water has gone away. It's not diluted now, it's concentrated. This process is known as distillation. If at all you are making something without distillation, that means it is not going to be concentrated. It is not going to be concentrated. So wine, whiskey, rum, brandy, which one do you think is not concentrated? It is obviously wine. Everyone can drink it. Whiskey, rum and brandy is a product of distillation. You do distillation to produce this, which is why the concentration of alcohol is way too much. Okay? Okay, ma'am. Next question. Activated sludge is a sediment deposited in. Activated sludge is a sediment deposited in. I'll make you do this question when we go ahead with sewage treatment. Okay, we'll come back and solve this question when we go ahead with sewage treatment. As of now, if I say, you will not understand it. Biogas is a mixture of, again this question came, biogas is a mixture of what? CH4, CO2 and also H2. Instead of H2S, it is H2. I told you, that's not a problem. Just give the correct answer. Now next question out there, match the column 1 with column 2 and select the correct options over here. So citric acid is produced by Aspergillus niger. Ma'am, that's a fungus. You told us. Yes, I told you. So Aspergillus niger, that is a fungus. Now what about butyric acid? Butyric acid is by Clostridium butylicum. Clostridium butylicum. So A goes with 3. A goes with 3. I have 3 options. And D goes with 2. D goes with 2. Also, I have 2 options. So basically, I need to know, I have a competition between this and this. I need to know what is the answer for this. Cyclosporin A. Cyclosporin A, my dear children, is a super important immunosuppressive drug. What is it? Immunosuppressive drug. If at all I want to make sure my immune system is not too active, I need to have it. Why do you think I want to make my immune system weak? Because suppose, my dear children, I am getting an organ transplantation. And if this organ is put in my body will react to it. And I don't want that reaction. At this particular point of time, I need to take some immunosuppressive agent. treatment, and that is cyclosporin A. And cyclosporin A is produced by a fungus, and that is Trichoderma polysporum. Trichoderma polysporum. This is an example of a bioactive compound. Okay, bioactive compound. Please remember it. And statin, which is there, this is there for stabilizing the blood cholesterol level, or basically lowering the blood cholesterol level. I always remember stabilizing because stable, statin, all of it sounds similar. Okay, so statin is there for stabilizing the blood cholesterol or lowering the blood cholesterol level, and basically it is produced by Monascus purpureus, which is also a fungi. Monascus purpureus, which is also a fungi. This is another bioactive molecule. So two bioactive molecules we studied. One is cyclosporin A, produced by Trichoderma polysporum, and statin, which is produced by Monascus purpureus. You know how you get to understand the names of these things in the chapter? By solving questions. That's the only way, I'm telling you. So then in that particular case, what will be your answer? Your answer will be this. B goes with 1 and C goes with 4, and rest of it we sorted. Right? Yes, ma'am. Now, I told you there's a question related to this. Which of the following is not an advantage provided by mycorrhiza to plants? Which of them is not an advantage provided by mycorrhiza to the plants, my dear children? Resistant to root-borne pathogens, it provides resistance. Increase in yield quality, no baba, I told you this is not the use of mycorrhiza. Tolerance to salinity and drought, yes, that is there. Overall increase in the plant growth and development, that is also there. That is also there. So the only thing that is odd one out is B. Cool? Yes. The microbial biocontrol agent for butterfly caterpillar is, the microbial biocontrol agent for butterfly caterpillar is what? Bacillus thuringiensis, Saccharomyces, Cyanobacteria, or Staphylococci. If at all you want to kill butterfly caterpillar, my dear children, you will use Bacillus thuringiensis. Why? Because Bacillus thuringiensis produces Bt toxin. Bt toxin, which gets activated inside the butterfly's gut, only inside the butterfly's gut. Why? Because inside the butterfly's gut you have an alkaline pH. In the alkaline pH, this toxin becomes active and can kill the organism. So the answer for this question is Bacillus thuringiensis. They come under biocontrol agents. Okay. So over here you have few more examples of all the biocontrol agent. We will go line by line, we'll study this, okay? Do not be worried, I have bigger questions, longer questions, which is why I'm not discussing that over here. Now, which of the following is a bacterial biofertilizer? Bacterial biofertilizer. Anabaena, Nostoc, Rhizobium, Oscillatoria. I told you this, and this. They come under cyanobacteria. Now, cyanobacteria also comes under bacteria itself, but here they are specifically asking about bacterial biofertilizers. The answer is Rhizobium. The answer would be Rhizobium. Okay? Yes. My dear children, if at all you are someone who is preparing for KCET and all of a sudden it has came. Okay, if at all you're someone who's preparing for KCET and is worried as in what to do, like where to start from, how to tackle everything, so ABHYAS KCET test series is right over here. So we have launched it early this time so that you can utilize it. As of now, I know it's a pressure of preparatory exam. Once you're done with that, make sure you go check it out and take it up. It is definitely going to help you out. Okay, that helped out a lot of children out there. It is going to help you out as well. Okay, so my dear children, we have completed the 1 marker, the most repeated 1 marker questions of microbes in human welfare, and now we'll go with the 2 and 3 marker questions, and here we will discuss more. I told you about sewage treatment and all, there are too many things, right? So I'll start off with the first question that we have, and that is for the sewage treatment, which is a long question. Okay, and with that we will be able to solve lots of smaller questions as well. So explain the stages of sewage treatment. How do you treat the sewage? Understand, first thing that we have is a settling tank. Remember, okay? In the settling tank, we have put everything that we want to separate out. Okay? This is known as the primary treatment. Now, there will be things that are going to float. There will be things that are going to sediment down. You remove it through filtration and you remove this through sedimentation. And this is your primary treatment. Whatever comes over here as a sediment, you call this as the primary sludge. The primary sludge. Now ma'am, so basically you're saying whatever has to be filtered, that got filtered, whatever had to be sedimented, that is sedimented. Now it's totally clean? No, it's not clean. We have to do secondary treatment. For that, we are going to take the fluid up over here. You call this fluid as what? The primary effluent. So in the primary thing, everything is primary. So primary sludge, primary effluent. So the primary effluent is now taken to the next tank. To the next tank, what do you call this tank as? This tank is super important, okay? This tank is known as aeration tank. That means here air is there. And air means there is oxygen also. There is oxygen also. So now the primary effluent is right up over here, okay? And in this primary effluent, I will be adding up some aerobic bacteria. Some aerobic bacteria, as in the one that requires oxygen. Apart from that, there will be some fungal filaments also. Apart from that, fungal filaments. So aerobic bacteria plus fungal filaments are there in the aeration tank. Together, both of them, I will call it what? What will I call it? Flocks. This is super. Ayo, that went. Just a second. That went. Okay, just give me a second. So anyways, first we were in the settling tank. Okay, and then we came in the aeration tank. Don't worry. I'm not going to make everything up there. Okay, aeration tank. Here I added two things. One is aerobic bacteria, as well as there is fungal filaments. You know what happens? They come together to form a mesh-like structure. You will now call this as flocks. And what is up over here? You have the primary effluent. You have the primary effluent. Now, when enough amount of oxygen is there, you know what happens? These aerobic bacteria are going to act on the organic matter present over here. So what exactly happens is that the organic matter which is there, it is going to get broken down. In the presence of what? In the presence of oxygen. So ma'am, you are saying there is lots of need of oxygen over here? Yes. What do you call this need as? Biochemical oxygen demand. The demand for oxygen is way too much when the organic matter is way too much. Okay, that means aerobic bacteria will also be way too much. So thing is very simple. If organic matter is too much, that means aerobic bacteria is also going to be too much. That means there is too much amount of BOD required. Oxygen demand is very much. If the amount of organic matter is less, that means aerobic bacteria is going to be less. That means biochemical oxygen demand is also going to be less. So we will work until the organic matter goes down. So until the organic matter goes down, the same process is going to be repeated. And the end, my dear children, we will find that the biochemical oxygen demand is reduced. And that's when we understand that everything is being cleared up. So who plays a major role in this? Flocks. What is flocks? Aerobic bacteria plus the fungal filaments. Remember it. Both of them super important. So from the settling tank, this was the first thing where we did the primary treatment. We took the things to an aeration tank. For the secondary treatment. Okay, from here starts the secondary treatment. Now what happens, my dear children? Over here I have treated everything. Again I am going to put it inside a settling tank. So this is the next time I am going to use a settling tank. What happens exactly over here? The aerobic bacteria, everything are going to settle down. The aerobic bacteria, everything is going to settle down. The treated primary effluent is going to be over here. Now you do not call it as a primary effluent, it's totally clean. You can send it to river, stream, wherever you want. But whatever is left over here, sedimented, what do you call it now? You will call this as activated sludge. Activated sludge, super important. What is this activated sludge made up of? Aerobic bacteria and the fungal filaments. Aerobic bacteria and the fungal filaments forms the activated sludge. That is important. So ma'am, what do I do with this sludge now? Listen, little part of it I can take it back and put in the aeration tank so that the next time something comes up, I can use this aerobic bacteria and fungal filaments that I took from here. In this case, I say that I have taken the inoculum. It acts like an inoculum, like a starter. Okay, it acts like a starter. Later it will divide in itself. Okay. Rest of it which is there, I am going to put into the next tank, the last tank out there, which is known as anaerobic sludge digester. anaerobic sludge digester. In the anaerobic sludge digester, this particular thing is going to get digested, my dear children. But over here, condition is maintained. Anaerobic condition is going to be maintained over here. That means anaerobic bacteria. Anaerobic bacteria are now going to feed on this aerobic bacteria and the fungal filaments. And what does it produce over here? It is going to produce the biogas. It is going to produce the biogas. This is the entire thing of sewage treatment. So you might be asked, what is activated sludge made up of? Please know it is aerobic bacteria plus the fungal filaments. Until when will you do this? Until the BOD level goes down. What is BOD? Biochemical oxygen demand. Okay, what is produced over here? Biogas. What eats what? Anaerobic bacteria feeds on aerobic bacteria and things like that in the anaerobic sludge digester. First the settling tank, then the aeration tank, then again the settling tank, and then you have what? The anaerobic sludge digester. If you want to take a screenshot of this, you can. This may go. Okay. Now, so primary treatment involves the physical removal of particles large and small from the sewage through filtration and sedimentation. Initially, floating debris is removed by sequential filtration and then by sedimentation. The primary effluent is passed into large aeration tanks where what is happening, my dear children? Useful aerobic things are being added up. It has been done until the biochemical oxygen demand goes less. And then BOD is the measure of, this question can be asked. If higher the BOD, what does it indicate? Organic matter is more or the waste is more. It is more contaminated. And then the whole thing that I explained to you, it's been put into points. I don't think we need to revise it again, right? Do we need to? I hope not. Okay. Now, we were talking about one question that I told you we will do later on. Let's come back to that question. Activated sludge is a sediment deposited in which tank? Settling tank, sedimentation tank, aerobic tank, and aerobic sludge digester tank. It is settling tank. It is settling tank. Okay? Easy now? Yes. Now we come back to this question that I was talking about. Describe briefly the primary treatment of sewage. We know primary treatment of sewage is the first physical step in wastewater treatment. The large and small solid particles are removed. Floating debris is removed by filtration. Grit, sand, soil, pebbles are removed by sedimentation. The settled solids form the primary sludge. The clear upper liquid is known as effluent. This effluent is sent for secondary treatment. What effluent? It is known as the primary form of effluent. Cool? Cool, ma'am. Okay, ma'am. What are flocks? Write their significance in sewage treatment. Flocks are what? Are the loose aggregates of aerobic microorganisms, mainly bacteria, along with what? The fungal filaments formed in the aeration tank during the secondary sewage treatment. What is their significance? They break down the organic matter present in the sewage. This reduces what? BOD. Lower BOD means reduced pollution potential to the effluent. They help in purifying sewage biologically before we release it. Right? So these are the uses. Okay? Yes, ma'am. Okay, so this came in model, MQP means model question paper. Now another question that came in model question paper is the role of fungi as biofertilizers. This also we discussed in the form of MCQ. So don't think that MCQs have to be taken lightly. While we study MCQs, that's also when we study certain concepts. So role of fungi as biofertilizers, it forms a symbiotic association with plant roots. That is glomus. Lives in close association with the roots of the higher plants, enhances the phosphorus absorption, increases the resistance and tolerance, resistance to pathogens, and tolerance to salinity and drought, and overall increase in the plant growth and development is being handled by this. There's mutual benefit to both fungus and the plant. Fungus receives organic nutrients from the plant as well because the plant is doing photosynthesis. So these are the things that we already discussed. I just put it down. Okay? Yes. Now mention the use of following bioactive agents, streptokinase and cyclosporine and statins. You know what? This question came in MQP1 and MQP2. Question number is 33. So, two of the things we discussed it. Are you able to recollect it? Cyclosporine A is what? Is something that is an immunosuppressive agent produced by a fungus. Remember the name? Trichoderma polysporum. Statin, stabilizing the blood cholesterol level or lowering the blood cholesterol level. Who produces it? Again a fungus, Monascus purpureus. Both of them are what? Bioactive molecules. My dear children, if at all you are not able to recollect, okay, the first thing that we need to do is, once I am done with this chapter, pause the video, okay, go through whatever. If you want to have a quick read of the summary of the chapter, do that and start referring to the PDF of this. Okay, the PDF of this, just refer to this. Or if you do not have the PDF as of now, just go back and read the questions one by one and then only go ahead with the next chapter. Listen, it's not just me teaching, it's also you being taught. Okay, you should also be able to understand it. And how do you know that? When you also go through each question and see it out. If at all you're able to answer it by yourself. So see that, check that, okay? So it's not just for views, it's for you to understand. So if that purpose is not being met, obviously this video is of no use, right? So please do that as well. Okay, so we were talking about that. Now we have streptokinase. Streptokinase, this we get from streptococcus, not staphylococci, Alexander Fleming. Streptococcus is a bacteria from where you get streptokinase. And what is this? This is a clot buster. Okay, it's a clot buster, as in any clot which is there in our blood, it can be busted with the help of streptokinase. It's an enzyme. It's an enzyme, my dear children, and it's produced by streptococcus. Whenever someone has heart attack or myocardial infarction, okay, that's when this particular thing is given. Used as a clot buster to dissolve clots in patients with myocardial infarction, that is heart attack. Okay, yes ma'am. These are the uses of bioactive agents, or these are the uses of bacteria in industries, medical industry to be specific. Next, name the microbes that help in the production of the following bioactive agents. Again, the same question in different way. Cyclosporine, Trichoderma, Polysporum. Here the names are asked, okay, not their function. Streptokinase, Streptococcus. Statin, it is Monascus purpureus. Okay. Next, explain the role of microbes in industrial products. So industrial products, starting from here. Aspergillus niger produces citric acid. What is Aspergillus niger? Fungus, ma'am, we studied it. Acetobacter aceti, producing acetic acid bacteria. Clostridium butylicum, butyric acid. Lactobacillus, lactic acid. Saccharomyces cerevisiae, ethanol, okay? And Penicillium notatum, penicillin, that is a fungus again. This is also fungus. Streptococcus, streptokinase. Trichoderma polysporum, fungus producing what? Cyclosporine. Monascus purpureus, fungus that produces statins. Please know it's a fungus, please know it's a bacteria, all of it. Okay? So over here, when you find similar names, that is a bacteria product. Aspergillus niger do not have any similar name with citric acid, so fungus. Saccharomyces cerevisiae does not have anything to do with ethanol, so fungus. Okay, yes. Now explain the role of microbes in household food products. So where does microbe play a very important role in household, my dear children? Lactic acid bacteria can be grown, they are grown in milk, and what do? They convert it into curd. So basically lactic acid bacteria, it breaks down, it partially breaks down the milk protein, which is casein. Which is what? Casein. So casein is a milk protein. It is partially digested by the lactic acid bacteria, which is why you have small clumps around there. Okay, and always remember, while it does that, it also releases vitamin B12, which is why there is an increased nutritional value of the vitamin B12. Now, baker's yeast, Saccharomyces cerevisiae, is used for making what? Bread. Okay, for making bread. So that's why you call it baker's yeast. There you called it brewer's yeast in brewery industry. Now, toddy's traditional drink is made by fermentation of sap from palms. Sap from palms. Apart from that, there is fermented fish, there is bamboo stalks and things like that as well. Apart from palms, we also use that. Now, large holes in Swiss cheese is produced due to production of large amount of CO2 by a bacterium named Propionibacterium shermani. And the Roquefort cheese is ripened by specific fungi, that is Penicillium roqueforti. The NCERT name is not given, so you need not study it. Just know it's a fungi which gives specific flavor. The Roquefort cheese is ripened by specific fungi which gives specific flavor. That's all. So these are the examples of household products, of microbes in household food products. Now, microbes as example of biocontrol agents. Biocontrol agents means basically we depend on what? Biofertilizers. We depend on we depend on pesticides for killing the pest. But suppose over here we do not have to depend on pesticides. Rather, there are organisms that will be feeding on other organisms. Yes, that will be a good deal, right? So the first thing that we have over here is predatory insects. Certain insects that kills the other insects. Ladybird beetle, you might have seen red and black spotted beetle. They feed on aphid. They feed on aphids. Dragonfly, they feed on Mosquito larva. So insects which are there, predatory insects, ladybird is there, it is feeding on aphid. Dragonfly is there, it is feeding on mosquito larva. Now microbial biocontrol agents. You have Bacillus thuringiensis. I told you it's a bacteria. Okay, this bacteria produces toxin. The toxin is inactive in bacteria's body, but when it gets inside the insect's body, in the alkaline pH of in the basic pH of the gut of the organism, that is the insect, it becomes active. The toxin becomes active, leads to perforation. So always remember, Bt bacteria or Bacillus thuringiensis is used for whose spores kill the butterfly caterpillars when eaten. Available as dried spores. Now this is a usual question that is being seen nowadays. It is available not as fresh spores but as dried spores mixed with water and sprayed on plants such as brassicas, cabbage, cauliflower and the fruit trees. Cabbage, cauliflower and the fruit trees. Now we have fungal biocontrol agents. For fungal biocontrol agents, whom do we have? We have Trichoderma fungi. It's a free-living fungi found in the root ecosystem, effective in controlling the plant pathogens. Okay, what is it? It's a free-living fungi. It's a free-living fungi. It is found in the root ecosystem. It is not dependent on anything. It's just staying there out there. And this trichoderma polycythemia, remember? So this trichoderma is saving the plant's root from other disease-causing agents. So it's a fungal biocontrol agent. Now we have viral biocontrol agent. The example is baculovirus. And it belongs to nucleopolyhedroviruses. So baculoviruses are there. It belongs to nucleopolyhedroviruses. They infect only specific insect pests and arthropods. They are all eco-friendly. They do not cause any harm. Okay, do not cause any harm up there. So yes, predatory insects, I hope it's clear. Microbial biocontrol agents, Bt. Fungal, you have Trichoderma, that is in the root of the plants. And viral, you have baculoviruses belonging to nucleopolyhedrovirus group. Yes. Now we have examples of biofertilizers. So nitrogen fixing proper bacteria is there, such as Rhizobium, Azospirillum, Azotobacter. Remember, Rhizobium which is there, it is going to be found in the roots of the legumes always. Always. They are not free living. They cannot sustain by themselves. They are dependent on it. So Rhizobium forms root nodules in the legumes such as pea plant. Fixes what? Atmospheric nitrogen into usable forms. So biofertilizers as in something that will increase the nutritional capacity of the soil. So over here this bacteria is helping in by fixing the nitrogen out there. And remember, they are found in the root nodules of the legumes. Now you have Azospirillum and Azotobacter. They are free-living bacteria that enrich the soil nitrogen content. Okay, they are free-living and they enrich the soil's nitrogen content. We have mycorrhizal fungi. That is the next one. We have studied a lot about it, glomus and everything. Yes, there we do not need to discuss much. Cyanobacteria, again blue-green algae. We have three examples: Annabena, Nostoc, Oscillatoria. Annabena, Nostoc, Oscillatoria. You have Uno. Like you play Uno, right? Here Ano is there. Annabena, Nostoc, Oscillatoria. They also fix nitrogen. They also increase soil's fertility and they are used as biofertilizers mainly in the paddy fields. Okay. So if at all you are asked what are the ones which are mainly used in paddy fields, it will be this. So please go through these examples. Super important. Yeah. Yes. Okay. Now, explain a typical biogas plant with a neat label diagram. So this is a typical biogas plant out there. What do you do over here? Listen, this is closed setup. Okay, this is a closed setup. Here you put the cow dung. Cow dung, my dear children, has some bacteria already in it. You call this as methanobacterium. Methanobacterium. They are anaerobic bacterium which are present in the gut of the cattle. In the gut, they help in the breakdown of cellulose, which is there in the plants. Okay. So that bacteria is there in the cow dung. You put the water, you mix it and you send it over here. Here you have a closed space. This anaerobic bacteria, they can grow and produce biogas. Now this biogas can be gone through this particular chamber to the homes, to different type of homes out there. Now whatever is left behind will be coming over here in the sludge, and this sludge can be used as fertilizers or manure. Okay, used as fertilizers or manure in the field. This is the thing. Okay, so please remember the diagram. And also it was developed in India by the effort of Indian Agricultural Research Institute, IARI, Indian Agricultural Research Institute, and Khadya and Village Industries Commission, KVIC as well. Okay, these are few things, please know about them. Now, explain the stages of sewage treatment. We have already done with it, which means that we are done with one chapter, microbes in human welfare. Okay, all the important questions of this chapter have been dealt with. Please, my dear children, pause the video, and if at all you want to solve the questions by yourself, do that. Cool? Yes, ma'am. Done, ma'am. Okay. Now we have biotechnology, principles and processes. You either get 2 1-marker questions and 1 5-marker question from this chapter. Starting with the 1-marker question right up over here. Before that, let me just drink a little bit of water. And you also, my dear children, please have some water. Stay hydrated. Okay. In the year 1963, 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA and the other added the group what? So basically, in the year 1963, inside the E. coli, you got 2 enzymes. What were these 2 enzymes? Remember, E. coli will have its own genetic material. Suppose a virus came and attacked the E. coli, that is a bacteriophage, came and attacked the E. coli, it will also put its genetic material inside. Now what will it do? Basically, this bacteriophage will try, the genetic material will try to get inside, incorporated inside the actual genome of the E. coli, such that it can use the host machinery for its own production. Which is why E. coli thought this cannot be happening. I am going to cut the genetic material right over here with the first enzyme, that is the restriction enzyme. The restriction enzyme. So restriction enzyme is going to cut this, okay, such that nothing can happen. Okay, but you should understand the restriction enzyme can act on its own genetic material as well. So you need to protect its own genetic material. How do you do that? By adding up some groups. By adding up some groups. You call these groups as methyl group. So you need an enzyme to add the methyl group. So that is also there. An enzyme is there that adds the methyl group up over here. Okay, so that it can remain protected. So in 1963, the 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA, that is restriction enzyme, and the other one added the group, which is methyl. Okay, yes ma'am. Next, so over here I have given the whole thing. You can go through it. Now, which of the following is the correct representation of primer annealing step in PCR? What is PCR? Polymerase chain reaction. You have a DNA over here, 1 from 3 dash, 1 from 5, here 5, here 3. First step that you do is separate them. That is known as denaturation, where you're gonna separate them. The next step that you have, my dear children, this is the first step. The second step is basically adding of primers. You know, the DNA polymerase basically needs to extend the whole line. You want to multiply, you want to amplify the amount of DNA, right? So first thing that you do is you add a primer. DNA polymerase cannot start off by itself. So you need to add the primers, in which end? The 3' end. This process is known as annealing. This is second step. This is known as annealing. Later, this whole thing is extended with the help of DNA polymerase. That is known as extension. Third step is extension. And basically, who does the extension? What is the name of the DNA polymerase over here? Taq polymerase. Taq polymerase. Remember, it is extracted from Thermus aquaticus, which is a bacteria that can sustain at higher temperature. Over here, the whole process is happening at high temperature. Denaturation at 95 degrees Celsius. This at basically 50 to 65 degrees Celsius. Over here at 70 to 75 degrees Celsius. So you need an enzyme that can remain stable at that particular time. So it is Taq polymerase. T for Thermus and AQ for aquaticus. Okay, so which of the following is the correct representation of primer annealing step? So over here at the 3' end it is being added, here at the 5' end, so it's wrong. Always primer attaches on the 3' end because DNA polymerase will be extending from 5' to 3' direction. So here it says 3' but here it again at 5', so wrong. Here at 5', wrong. Here at 3' and there also 3', this is correct. So your answer for this question becomes D. Okay, DNA replication happens in 5' to 3' direction. Cool, ma'am. Next question. The chemical nature of the primer used in the polymerase chain reaction is? The chemical nature of the primer. The primer that we are going to use over here in polymerase chain reaction, my dear children, how will it be? Is it made up of RNA? Is it made up of DNA nucleotides? It is usually DNA nucleotides. So in general, when replication is happening inside our body, at that particular point of time, it's usually RNA nucleotides which are being used. But here, in this particular case, anyway, we just want a new strand up there, and we don't depend on our human machinery, right? So we can have the DNA strand, okay, the small nucleic acid. So it is deoxyribonucleotides that we use. Not ribonucleotides, it will be deoxyribonucleotides. Okay? Yes, ma'am. Okay? It's not sides. Deoxyribonucleoside means basically just sugar is there and the nitrogen base is there. Here you need phosphate also. In that case, you call it deoxyribonucleotide. This whole thing is nucleotide. Okay. Done? Done. Now, in the given diagram of PBr322 vector, identify the restriction sites A, B, and C, and also D. Always remember this particular picture. This picture should be there in your head even when you sleep. Okay. So, one origin of replication site over here, one ROP site over here. ROP is a protein which is super important for replication to happen. You have two antibiotic resistant gene. One is ampicillin resistant gene over here, and one is tetracycline resistant gene over here. Okay. And now, my dear children, you need the enzymes. These are the sites which you have. Now, there are sites where the enzymes will come and cut. So, what are the sites? Over here, the first site is occupied by PVU2. PVU2. This is an enzyme, restriction enzyme, and its cloning site is this, the site where it cuts. Here you will be having two of them. One is BAMH1 and the other one is SAL1. BAMH1 and SAL1. Here also you have two, PST1 and then PVU1. So if at all you see, both the sites is just 1, 1. Here you have PVU2. Okay, and here you have three of them. One is the very famous EcoR1. Okay, one is the very famous EcoR1, and then you have CLAV1 and you have HIND3. CLAV1 and HIND3. So one dundee over here, one dundee over here. Listen to me carefully. 1, 2, and 3. Right? 1, 2 of them are there, and then 3 of them are there. You can remember like that. And the third one ends also with 3 and also ends with 3. But over here, the first one does not start with 1. It starts with 2. So PVU2, BAMH1, SAL1, PVU1, PST1, EcoR1, CLAV1, and HIND3. Remember that. Okay. So over here, what is A? A is origin of replication, without which replication cannot start off. What is C? It is tetracycline resistant gene. What is B? It is ampicillin resistant gene. And what is D? It is HIND3. Okay. So A, K, yes. So your answer for this question becomes B. Okay. This picture, you might be asked to draw it or identify it. You might be saying that if at all I am cutting with BAMH1, which gene will be not doing its function? It is tetracycline resistant. It won't be tetracycline resistant anymore because you're cutting that particular region out there and you're inserting something. Okay, restriction site of EcoR1 enzyme is. What is the restriction site of EcoR1 enzyme, my dear children? You know, HIND2 was the first restriction enzyme to be discovered, and it has a site consisting of 6 nucleotides. Similarly, over here also you have 6 nucleotide pair, and the site is 5GAA-TTC. It cuts between G and A. So it is 5 dash, it's not 3 dash, it is 5 dash GAA-TTC 3 dash, and it cuts between G and A. Okay? Yes, ma'am. So when it cuts, okay, it makes cut like this. See, over here I'll explain you the concept. So it was GAA-TC on this side also. Always remember, restriction site are going to have palindromic sequence, same type of sequence. If you read from here, it's going to be the same. So I made a cut between G and A, I made a cut between G and A over here, I got a L-shaped thing. Similarly, from on this side also, I'll make the same thing. Now, wherever I want to insert it, I will be cutting the plasmid also with the same type of enzyme so that I get the same sticky ends. These ends are known as sticky ends. Now this part can come and join over here, right? and the other part which is there will also be coming and joining on the next part of the plasmid. So that is known as sticky ends. They're waiting for hydrogen bonds to be formed. Okay, question number 6. Fill in the blanks by choosing the appropriate words. Morphine, endometrium, reserpine, fossils, vector and myometrium. The plasmid DNA acts as dash to transfer the piece of DNA attached to it. The plasmid DNA which is there acts as what? Acts as a vector. Acts as what? It acts as a vector to transfer the piece of DNA attached to it. Vector as in a carrier. Okay, always remember plasmid DNA is a vector. Okay, plasmid DNA is a vector, but not all vectors are plasmids. You have different type of vectors. Okay, plasmid acts as a vector, but not all vectors are plasmid. Now, which of the following is not a feature of plasmid? What is plasmid? First of all, in E. coli, you have your own genetic material, everything which is coding for a protein which is going to help in the sustenance of E. coli. Apart from that, you have an extra chromosomal region. It is self-replicating, okay, and basically it is double-stranded, it is circular structure, and it is extra chromosomal. It is not producing any protein, which is very important for this particular organism to survive. Okay, so which of the following is not a feature of plasmid? Extra chromosomal, correct. Circular structure, correct. Single-stranded, wrong. It is double-stranded. Independent replication, correct, because it has origin of replication. Now, Agrobacterium tumefaciens is used as a vector for cloning genes in. Agrobacterium tumefaciens is a bacteria. And now why do you need this organism? Because it acts as a vector. Its plasmid acts as a vector for plants. So Agrobacterium tumefaciens, my dear children, it has an extra plasmid out there. You call this plasmid as TI plasmid, tumor inducing plasmid. Ma'am, it induces tumor? Yes. How? Basically, it has a region which is known as transfer DNA. It is known as transfer DNA. Now, when this bacteria goes and affects a plant, it transfers this part. And this part is responsible for forming tumor in the case of plants. Now, suppose I am removing the transfer DNA part. I am removing the transfer DNA part. Ababa, everything got removed. Okay. And I am inserting my gene of interest, the gene that I want. Anyway, the bacteria, what do you say, has the same function. It goes and affects, and while it affects, my dear children, it passes on the transfer DNA into the plant. It not transfer DNA, the gene of interest into the plant. That is why it is going to act like a vector. So Agrobacterium tumefaciens is used as a vector for cloning genes in the case of plants. In the case of animals, what do we use? In the case of animals, we use retroviruses. Retroviruses, you know, HIV virus affects our human being because it knows how to affect us, right? Similarly, if we use the same machinery for transferring something important, we can. So retroviruses are something which are used as vectors in the case of animals. Yes, ma'am. Done, ma'am. So over here, yes. Agrobacterium tumefaciens transforms normal cells into a tumor by delivering a piece of DNA known as, what do you call this DNA? Agrobacterium tumefaciens transfers a normal cell by transferring something very important. What do you call this as? tDNA, that is transfer DNA. I told you that causes the whole trouble. Now, which of the following is the correct order of processing of PCR? Annealing, denaturation, extension. Arey Baba, you need to denature first, then you have to attach the primers, then you have to extend it. So your answer for this question becomes C. Thank you, ma'am. Welcome, dear. So statement 1. Sticky ends are resulted. Anyway, you guys are not going to say thank you to me. Are you? Are you? Well, you do not need to say thank you to me, just that, my dear children, the efforts that I am putting up over here, you know, it needs a lot of effort to come over here, stand in front of the camera, talk to yourself. It feels like talking to yourself, right? And still maintain the energy. So we are doing that for you guys so that you do not get bored at any point of time. Okay, so we need the same amount of energy from you guys. When you feel that you are not able to do it, trust us. You are more capable than you think. Go ahead and do it. Okay, so if at all you're feeling sleepy now, wash your face and come back and study, because it's important for you to score good. Yes, so sticky ends are resulted due to the action of restriction enzymes. Is it correct? Restriction enzyme cuts, that's why you get sticky ends. I told you the photo, I showed you. Yes, ma'am, correct. Stickiness facilitates the action of DNA polymerase. So now sticky ends are there. Now when the portions are joined together, do you think that is going to help anyway in the action of DNA polymerase? DNA polymerase is going to extend, right? For chipkawing things, for sticking things, do you need DNA polymerase? No, Baba. It should have been DNA ligase. So even though the sticky ends stick to each other, but the thing is we also need DNA ligase to make the bonds and everything very, very clear for making the phosphodiester bond. So it's DNA ligase, not DNA polymerase, which is why the statement would be wrong. So statement 1 is true, but the statement 1 is true and statement 2 is what? False. Okay? Yes, ma'am. So over here the sticky ends are being mentioned. So I cut the foreign DNA and the what you call vector DNA. I made the sticky end join, but still you will be needing what? DNA ligase to form the exact bonds out there. Okay? The purpose of stirred tank bioreactor is, why do you need a stirred tank bioreactor? Basically, stirred tank bioreactor makes sure that oxygen is being mixed thoroughly, everything is being mixed thoroughly, everything actually should be mixed thoroughly, but it makes sure oxygen is something that is being given enough, because aerobic bacteria might be the one which are cultured and they need oxygen to survive. So the purpose of stirred tank bioreactor is providing anaerobic condition. Arre Baba, no, you need aerobic. Purification of the product, no. Addition and proper mixing of preservatives, that is there, but more than that, availability of oxygen throughout the process is the major thing. So D will be your answer for this question. Okay, yes ma'am, done. So this is stirred tank picture, stirred tank bioreactor picture. This is super important. Listen, so over here your acid base for pH control is there, so that you understand whether the pH is maintained or not. There is a steamer for sterilization. Once it is whole being done, you pass the steam to sterilize it. Okay, there is a motor so that the impellers can move. These are the impellers that can move, my dear children. There's a foam breaker at the top. Now you can get questions like, hey, what is this? This is a foam breaker. Okay, it does not allow foam to form. If foam is being formed, the whole process gets ruptured in some certain ways. So for stopping that, you need a foam breaker. This is the culture broth in yellow, and this is sterile air that is being put over here. Okay. Next, identify the correct statement. Okay, let me identify. HindII cuts the DNA at specific sequence of 6 base pairs. HindII is the first restriction enzyme. So in E. coli, you got to understand that there was restriction enzyme, but you could not discover it. The actual thing that you discovered was HindII. It is from, always remember, the name comes, the first letter from the genus name, the second 2 letters from the species name, and this basically from the strain. And this would be the discovery number. Okay, discovery number, the order of discovery. So HindII is something that was discovered first, and it has a base sequence of 6 base pairs. Microinjection is the technique of introduction of rDNA into the plant cell. Microinjection. So there are certain techniques where you need vectors for transferring things, such as agrobacterium tumefaciens, or you can go vectorless also. One such vectorless method is microinjection, where what do you do? Basically, there's a cell and there's a nucleus. You inject inside the nucleus the DNA that you want. But do you think it can be done in a plant cell? Plant cell has an additional wall which is known as cell wall, and you cannot do that here. So microinjection is a technique of introduction of rDNA into the plant cell will be a wrong statement. You cannot do that. It is used. This is used as a vectorless method in the case of an animal cell. Animal cell. Now biolistic is a technique of introduction of rDNA, recombinant DNA, into an animal cell. Now my dear children, in an animal cell, suppose this is an animal cell, does not have a cell wall. And what happens in biolistic, it is also known as gene gun method, where you have a gun-like thing and you insert the DNA inside the nucleus with the help of filaments of gold and tungsten. Why do you need to do that over here? You can just do microinjection rather, right? So biolistic is a method that you apply in plant. Remember this name, you might forget it. Biolistic, gene gun method, everything same. Okay. Now, retroviruses are commonly used cloning vectors in plants. Retroviruses are commonly used cloning vectors in the case of plants. Is it? It is animals, right? It is animals. Retroviruses affects the animals. Agrobacterium tumefaciens is the cloning vector in the case of plants. So, your only answer for this question becomes A. Are you? A. And over here, I have the explanation of all of it. So, if at all you want, you can read through it. Okay, now we have come to the 5 marker questions of this particular chapter. So who all needs a little bit of water? I need a little bit of water, so I drink, and when I drink, I remind you also. Please stay hydrated. It helps you. Okay, let's go. Explain the process of preparation of competent host for transformation with recombinant DNA. What? So explain the process of preparation of competent host for transformation with recombinant DNA. That means, my dear children, over here suppose I have a box, and then over here I have the E. coli, and I also have the recombinant DNA up there, the new combination of DNA out there with the plasmid. Okay, I want to make sure this goes inside this. The recombinant DNA goes inside this. Well, this process of making sure that the E. coli takes it up is known as to make the host cell competent, compatible to take it up. So explain the process of preparation of a competent host for transformation with recombinant DNA means to make sure that this guy will be able to take it. How do you make it sure? You need to do a process. What is the process? First of all, you're going to treat the bacterial cells with divalent cations. Divalent cations as in whose valency is 2. So calcium is a cation whose valency is 2. So you can take calcium chloride, or basically which has calcium. So first thing, bacterial cells are treated with divalent cations to make them competent. How does that help in making them competent? So calcium ions neutralize the negative charges on the DNA and the cell membrane. So it's not just the E. coli which is treated with calcium, but also the DNA. Both of them should be neutralized. Remember, DNA is negatively charged. Your cell membrane has a different charge altogether. So if at all, it has the same charge. So it won't be able to get inside. With the same charge, it will be repelling down. So what do we need to do? We need to make sure that it is neutralized. We need to make sure that it is what? Neutralized. So the calcium ions neutralize the negative charges on DNA and cell membrane, increasing the permeability. Now, basically, neutralization is being done. Both of them are ready to take them up, but you need some space to get inside. How will you get inside the E. coli? For that, first of all, the E. coli is put in a cold medium. The second thing that is being done is heat shock treatment. All of a sudden, it is being transferred to somewhere where it's heated. Okay, the temperature is being shifted. And when this temperature is being shifted, it creates transient pores, as in temporary pores. When this temporary pores are there, obviously this thing can enter inside. The recombinant DNA will be able to enter inside. Again, you need to close these pores. What do? You put it inside cold medium. So cold, then heat shock, then cold. You do that, and then, my dear children, everything is sealed up. The mixture of cells in the DNA is then given heat shock. So before that, you need to treat it with cold medium, then heat shock, 42 degrees Celsius for 90 seconds, and then cooled on ice. This creates temporary pores, allowing recombinant DNA to enter the bacterial cells. Okay, done. This is how you make the cells competent. Now the next part of the question. Refer to the given diagram of PBR 322 and answer the question that follows. Okay, mention the restriction enzymes that can be used for insertional inactivation of antibiotic resistant gene. What? So I have to talk about the restriction enzyme that can be used such that there is an inactivation. There's an inactivation of the antibiotic resistant gene. You have two antibiotic resistant genes right over here. What are those? One is ampicillin and the other one is tetracycline. If I want to make this inactive, I will be either using BAMH1 or SAL1, because when I use it, obviously it is going to make a cut over here, and I'll insert my gene of interest, which will inactivate this tetracycline. This is known as insertional inactivation. So I can either use BAMH1, SAL1, or I can use PVU1 and PST1 to basically do insertional inactivation of ampicillin resistant gene. So these are the options that I have. So you need to write that. Okay, you need to write that. BAMH1, SAL1 or, okay, wait. BAMH1 or SAL1 can be used, and over here PST1 or PVU1 can be used. Not PVU2, it's just PVU1. Inactivate ampicillin, and that inactivates tetracycline resistant gene. Now, How many cloning sites are present in the given PBR 322? How many cloning sites are present, as in how many restriction enzyme sites are present is being asked. So 1, 2, 3, 4, 5, 6, 7, 8. 8 of the restriction enzyme sites are being present. Number of cloning sites in PBR 322 is what? 8 unique restriction sites. Okay. Yes, ma'am. Done. So this is MQP1 question, very important. Now, next question. Give reasons for the following. Restriction enzymes are called molecular scissors. Why do you think restriction enzymes are called molecular scissors? Because, my dear children, they are responsible for cutting the DNA, the molecules out there. That is why it is known as molecular scissors. You have two types of restriction enzyme. One is endonucleases that cuts within the DNA. One is exonucleases that cuts at the ends of the DNA. Now, DNA ligase, that is the first point. Okay, so we'll read it out once. Because they recognize and cut DNA at specific base sequences, recognition sites, just like scissors cut at precise points. Give an example of EcoR1. It cuts at what site? 5GAA TTC 3'. It cuts between G and A. Mention that. Okay. Next, DNA ligase is used in genetic engineering. I told you, you need to make phosphodiester bonds between the two. Whenever you make recombinant DNA out there, you need to join the bonds out there between the nucleotides. You need to make the new phosphodiester bond. For that, you need DNA ligase, because it joins the two DNA fragments by forming phosphodiester bonds between the sugar and phosphate backbones, helping to seal the foreign DNA into the vector. The foreign DNA into the vector which is going to carry this. Now, chilled ethanol is used in DNA isolation. So, in DNA isolation method, no. So, suppose in the cell, lot of things are there. RNA is there, protease is there, proteins are there, lot of things are there. I basically separated everything and I just have the DNA now. But I need to make sure, or basically I digested the proteins out there, I digested the RNA out there, the DNA is intact. But how do I separate it from the RNA and the protein? You need to add chilled ethanol. When you add chilled ethanol, what happens, my dear children? It floats around. Okay, it will be floating around. So over here, because DNA is insoluble, because DNA is what? Insoluble in cold ethanol, so it precipitates out from the cell extract, making it visible and easy to collect. That is the reason. Now, smaller DNA fragments move farther in a gel slab. Why do you think smaller DNA fragments move farther in a gel slab? If you take an electrophoresis chamber, always remember you have cathode over here, and cathode is negatively charged, and you have anode which is positively charged. Anion is negative, but anode is positive. You have DNA being put inside the wells over here. Now, DNA is negatively charged, both of them ripple, they will be moving. So, the smaller ones are going to move really fast compared to the bigger ones. Smaller ones move faster than the bigger ones. Why? Because they have less resistance. Okay, so always remember one very important thing. In the gel electrophoresis chamber, DNA does not go on the top, do not move on the top of it. Okay, it shows sweeping effect. Sweeping effect, as in it goes through the gel. It will be passing through the gel. Okay, it shows sweeping effect out there. You need to remember that. Okay, so over here, smaller ones will find less resistance, whereas larger ones will find more resistance. Smaller DNA fragments move farther in a gel slab because of the same reason. Okay, and also remember one more very important question. Who separate first? Bigger ones or smaller ones? Arey, we do not know. Anyone can separate. That is not a matter of concern. Anyone can separate. So if at all it is said smaller ones separate first, it would be a wrong statement to say. We do not know about it. So because during gel electrophoresis, smaller fragments face less resistance from the gel matrix and therefore migrate faster and farther towards the positive electrode. Okay. Now, thermostable DNA polymerase is used in polymerase chain reaction. Why? Because my dear children, DNA polymerization happens at a very high temperature in the case of PCR. So that's why you need thermostable polymerase. So because enzymes like Taq polymerase can withstand the high temperature used to denature the DNA strands, allowing the PCR cycle to continue without the enzyme breakdown. So enzyme should not break down, and for that obviously you need to have a thermostable DNA polymerase. Here I mentioned about extension also. Okay, yes. So here chilled ethanol, when it is added, you can see the DNA precipitates out. Gel electrophoresis, the DNA content is being moving. and the smaller ones move faster. Here the DNA ligase is acting like a glue. Cute glue, right? Yes. Now, just give me a second. Let me see how many questions are there. Okay. Genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. Genetic engineering is a core technique of biotechnology. In the background of this, complete the following table. Gel electrophoresis, A, visualization of DNA. So, gel electrophoresis, DNA polymerase is a tool that we use, and what do we use to visualize it? Oh, wait. Sorry. Over here, my brain is not braining as of now. So, gel electrophoresis is there. Visualization of DNA fragment is there. In gel electrophoresis, what do you use? What do you use, my dear children? That is the question. Ethidium bromide. For what? Visualization of DNA fragments. And ethidium bromide gives a bright orangish red color, and also remember it can only be viewed under a UV light. So even though you put ethidium bromide, you won't be able to see the DNA until you treat it under the UV light. Okay, so your answer for this question would be ethidium bromide. Over here you have DNA polymerase and C is there. So why do you need DNA polymerase? You can use PCR over here. In PCR, DNA polymerase is used for what? Amplification of DNA. Amplification of DNA. Now here gene gun is given. Gene gun is biologistic method. So gene gun and then dash and then transfer of recombinant DNA. Gene gun and then dash. What will be your answer for this? What do you use in the gene gun for the transfer of DNA? It is the filaments of gold and tungsten. You cover it with DNA and that's how you transfer it. And then you do the gun, the gold and tungsten goes inside along with the DNA. So that's the tool out there. Now isolation of the DNA, what is the tool that we use? Remember, this is for tools. Isolation of DNA, what do we use? Chilled ethanol, and that helps in precipitation of the DNA. So over here you have the list of things. Please go through it. Ethidium bromide for saying, and this gives a bright orangish red color, and polymerase chain reaction is there. It helps in amplification of DNA. Gene gun uses tungsten or gold particles coated with DNA, and isolation of DNA is done with the help of chilled ethanol. Okay? Okay. Next, given below are the diagrams of plasmid A and B. Okay. See the plasmid. So this is PBR 322, but this is something else. This is having lag-Z gene. Lag-Z gene, my dear children. Okay. Which plasmid is or are you select for cloning and why? Which plasmid will you select for cloning and why? What is insertional inactivation? And then will the number of culture plating you should make is the same or different to select the recombinants? Okay. If insertional activation is possible. Oh my God. Too much complicated. Let's make it easy. First of all, which one do you choose? Generally, you will choose this lag-Z gene. Why? Listen to me carefully. This is a plasmid. This plasmid has a gene that is lag-Z. And lag-Z, my dear children, lag operon you have studied, right? It produces beta-galactosidase. And beta-galactosidase over here, when you add a substance such as chromatin, some chromatin, that gives color. Okay, for example, the substance that you usually take is X-gal. When you add it, this thing is being treated and it gives a blue color. It usually gives what? A blue color. Now, my dear children, listen. If at all I am putting my gene of interest over here, then this lag-Z gene is going to get inactivated. And will it produce beta-galactosidase? No. So basically, will it give blue colors? No. So even if X-gal is there, it is going to give white color. White color colonies. So the bacteria that has this particular plasmid with my recombinant DNA, okay, will be giving white colonies. The bacteria that does not have my recombinant DNA will be giving blue color colony. So it is very easy for me to find it out with the help of this particular plasmid. And what is insertional inactivation? When I am inserting something and because of that, the property of that particular gene is being lost, this is known as insertional inactivation. Now this is a question. Is the culture plating the same in both these cases? So over here, when you did this in the same petri dish. Okay, in the same petri dish. So somewhere in the same petri dish, the organisms that had my plasmid, my dear children, it gave me white colonies. The organism that did not have my plasmid, recombinant plasmid, it gave me what? The blue colonies. So this can be performed in one single petri dish. Only one plating, single plating is being required. Now I can take these organisms and do my experiment because these are the recombinant ones. But when you talk about PBR 322, things are quite different. Why is it different? Listen to me carefully. So in PBR 322, first thing that we need to understand is that whether the plasmid, okay, whether the E. coli which is there has taken up any plasmid. So listen to me carefully. There will be some plasmid. So I'm making 3 scenarios. Or do you want me to make in a different, I'll make it in a different area. Okay. I have 3 plasmids. Okay. This plasmid has both ampicillin and okay, or I'll say this is wait, I'll remove it altogether and I'll make it over here. This is ampicillin resistant, and over here you have tetracycline resistant gene. Okay, this is having ampicillin resistance, and over here you have the gene of interest. Gene of interest I'll show by blue. Which one of them is the recombinant plasmid? This one is the recombinant plasmid. Recombinant plasmid. So first thing that I am going to do is basically I am going to find out the E. coli that has taken up plasmid. So it can take this plasmid, it can take this plasmid, or it can take no plasmid also. How do you understand whether the plasmids are being taken up? So this is my recombinant plasmid. I do not know that this plasmid has not been recombined. I do not know that. Okay. Anyway, first thing that I want to know is basically which one of the E. coli has taken up plasmid, any plasmid out there. So in that case, what do? I'm going to treat it with ampicillin first. When I treat it with ampicillin, this is able to survive. Why? Because it has ampicillin resistant gene. This is also able to survive. Why? Because it has ampicillin resistant gene. Will this particular E. coli survive? No. So this is what? A non-transformant. This is a non-transformant. Transformant means it has not taken, non-transformant is not taken up anything. It has not been transformed. These two are transformants. These two are transformants. Now, what is my role? My role is to find what are the recombinants, the ones that have my gene of interest. So for that, we need another plating. So now we have another plating where what do? Again, E. coli is there with this one, and E. coli is there with this one. This time, I am going to treat it in tetracycline medium. Which one will survive? The one that has tetracycline resistance, that will survive. This one is not going to grow. This one is not going to grow. So which one is the recombinant? Why is it not going to grow? Because it is insertionally inactive, because it has my recombinant plasmid. So which one is the recombinant? Actual sense. Which one is a recombinant in between both of them? Both of them are transformants. But which one has the recombinant one? It is this one. The one which is not able to grow is the recombinant. And the one which grows, my dear children, it is a non-recombinant. So I am going to take the one which is not able to grow. I am going to take the one which is not able to grow. So you need 2 cycles of plating over here. 2 plating is required over here in this particular case in PBR 322. First is to find the transformant, second is to find the recombinants. Okay, so which plasmid will you select for cloning and why? Plasmid B is selected. Why? Because it has LAG-Z gene which helps to easily find the recombinant colonies. When a foreign DNA is inserted into the LAG-Z gene, the gene gets inactivated. This allows blue-white screening. White colonies recombinant, blue colonies non-recombinant. Now, what is insertional inactivation? It means the gene stops working because a foreign DNA is inserted into it. In plasmid B, the LAG-Z gene stops producing the enzyme beta-caroteridase after insertion of the foreign DNA. Now, will the number of culture plating be same or different? Comment. So, obviously, the number of plates will be different for both. For plasmid A, needs 2 plates, one with ampicillin for finding the transformants and one with tetracycline for finding the recombinants. For plasmid B, needs only 1 plate, white colonies give recombinants, blue one non-recombinants. Okay, that's all. This question is a bit complicated, but you should know how to tackle this as well. Now, study the diagram given below and answer the questions that follows. What is EcoR1? EcoR1 is a restriction endonuclease. It is a restriction endonuclease which cuts the site 5GAA TTC 3-. Now, how is the action of exonuclease different from that of endonuclease? Endonuclease cuts within. They break down the sugar phosphate backbone within the DNA, not towards the tip. So that is a difference. So exonuclease cuts the DNA from the ends, whereas endonuclease cuts the DNA within the strand. Exonuclease removes nucleotides one by one from the terminal end. Okay, from the terminal end. Endonuclease break the DNA at specific recognition sequence. Exonuclease's example is enzyme that trims the DNA ends. Example is EcoR1, Hint3, all of it. Okay, and over here, what is EcoR1? It's a restriction endonuclease enzyme. Recognizes specific palindromic sequence and cuts between G and A, producing what? Sticky ends. Now there is one more question. How are sticky ends formed on DNA strands? Why are they called so? So how are sticky ends formed like this? They are going to be formed like this. Do I have an answer for that? Yes. Sticky ends are found in restriction enzyme such as EcoR1 cuts the DNA staggeredly, okay, between bases G and A. This produces single-stranded overhangs with complementary sequence. They are called sticky because these ends can easily pair with the complementary base of another DNA, helping in joining the foreign DNA with the vector DNA. Also mentioned, the vector DNA is also being cut with the same enzyme. That is also important. And please make sure you at least draw some part of this particular thing. Okay, I'll zoom this in. Okay, TTC, just make vector DNA, foreign DNA, make a cut over here. In opposite L shape it should come. And then join both of them. Now, write 3 vector-free techniques that can be utilized to transfer recombinant DNA into a ready host cell. Microinjection, biolistic, and then the heat shock method. Write 3 vector-free techniques, those are the methods. Agrobacterium tumefaciens acts as natural vector for cloning genes in plants. Substantiate, you have to explain all of it. We already spoke about it. It has tumor-inducing plasmid, you have tDNA, you remove that tDNA, put the gene of interest, and then you do. Over here, microinjection, biolistic, and electroporation is given. So electroporation is another method where basically you give electric impulses so that it creates pores and the DNA can be taken up. That is also a vector-free method. Or else you give heat shock treatment, that's all. Yes. Write a labeled diagrammatic representation of recombinant DNA technology. So how do you do that? You do like this. So foreign DNA, vector DNA is taken. Foreign DNA and vector DNA both are cut with the same enzyme, and now you join it. You see, the cut is there. You put it over here, and basically you join with the plasmid, and the plasmid is allowed to transform the E. coli. In the E. coli, there is recombinant and non-recombinant. That's all. Okay. Yes. Now, steps involved, the same thing that we discussed. I have put it over here, and I have explained. That's all. There's nothing new. Yeah. This step is super important. You might get questions on the basis of this as well. How do you isolate the DNA? So there are different organisms, no? You know, bacteria. Bacteria have cell wall which is made up of peptidoglycan. Peptidoglycan. Oh shit. Oh my god. Sorry. So over here I have peptidoglycan. So it has to be broken down with the help of lysozyme enzyme. So I will treat it with lysozyme enzyme to get inside the cell of a bacteria. Now if you're talking about plants, plant cell wall is made up of cellulose, for which you need what enzyme? You need cellulase. Okay, you are going to need the enzyme that is cellulase to break it down, cellulose. Now next, fungi. Fungi membrane is made up of chitin, and for breaking down chitin, what do we need? We need chitinase. We need chitinase. Okay. Yes, ma'am. So briefly explain the steps of recombinant DNA technology. The first step is isolation of DNA. This is what you do, and basically after that you also treat it with RNase to break down the RNA once you get inside the nucleus, and you also take the help of protease to break down the proteins, and finally use chilled ethanol to do the separation. Okay. Now the last step is also something that we have not discussed, downstream processing. Downstream processing means separation and purification of our component. Protein, whatever thing we need to separate it, we need to purify it. That is known as downstream processing. Later it is treated with what? Preservatives, and it is made to undergo quality testing as well. All of it is also being done. Cool? Yes, ma'am. Fine? Okay. Now we go ahead with the next chapter, that is biotechnology and its applications. Again, my dear children, take a choo-choo break, go through the entire chapter, and then let's go ahead with the next chapter. And just give me 1 second, I'll also be going on a short break. Okay, so let's go ahead with biotechnology and its application. Over here, I have the first MCQ question. Given below the steps in the gene therapy of patient with adenosine deaminase deficiency. Adenosine deaminase deficiency, okay? And what you have to do, which of the following show the correct order that those steps must occur? Okay, anyways, what is the correct order of the steps that is being asked? So my dear children, first of all, adenosine deaminase is an enzyme, okay, which is found in every cell. And what does this enzyme do? Basically, it makes sure that the cell is toxic free. Okay, it detoxifies the cell. Now, certain people do not have the gene that codes for the enzyme. That means there is no enzyme. That means there is toxic buildup in certain cells. So that's a big problem. This toxic buildup is a big problem for certain cells, that is lymphocytes, your immune cells out there. So you need to do something with to the lymphocytes out there. Otherwise, how will you provide yourself immunity? So we have to do something, and we came up with a solution, and that is gene therapy. So what do we do? Suppose I am the person. So from me, lymphocytes are being taken up. The first step that is being done is lymphocytes are being taken up. Then these lymphocytes are cultured, my dear children, so that we can increase their number. Okay, so first step is lymphocytes are collected from the blood of the patient. 3 is the first one. Then lymphocytes are grown in a culture outside the human body. And then, my dear children, I am going to make sure that the retrovirus, okay, retrovirus carries my gene of interest and insert my gene of interest into all of these cells. So I am going to take the help of retroviruses to basically insert my gene of interest into the lymphocytes. So lymphocytes are altered with a functional ADA gene. Done? Yes, ma'am. Now, everything is clear. What we do, we take these cells and put it back inside my body. Inside my body. So genetically modified lymphocytes are infused back into the patient. That is the next step that we do. So basically we have 3, then we have 1, and then we have 2, and then we have 4. So your answer for this question would be 3, 1, 2, 4. C. Okay, clear? Always remember, what are you going to insert? You are going to insert the cDNA, complementary DNA. My dear children, you have DNA. The DNA undergoes transcription to form what? RNA, right? The DNA undergoes transcription to form the RNA. This is how things work. Now, over here, if okay, now this DNA which is there, it has coding sequence and non-coding sequence, and that is the exact thing that comes in the RNA also, coding and non-coding. This RNA, which is mRNA, will be made sure that it just has what? Coding sequences. Non-coding sequences are removed in the mRNA. So now we have everything that is needed for making adenosine deaminase out there. Why not do reverse transcription of this and make a DNA which does not have the non-coding sequence? Yes, that is being done, and that is the cDNA, complementary DNA. Okay, the mRNA which does not have the non-coding sequence, you do reverse transcription of this and you get complementary DNA, and that is what you insert with the help of retrovirus. Just for your understanding, I am telling you, I do not know if at all a question could come up or not. Okay, just for your understanding, have this in your head. So this is gene therapy. This was done in a 4-year-old patient, my dear children, who had a deficiency with adenosine deaminase. And you know what? She is still there. Go check it out when you are free, not now. So the plants developed by the fusion of protoplast of 2 different plants are called. Okay, so over here I have a plant cell, I have another plant cell. I told you the plant cell are very unique because they have what? They have cell wall. They have cell wall, right? Suppose I am removing the cell wall. Suppose I remove the cell wall. What will you call this now? This is a protoplast, and this is another protoplast. I have 2 protoplasts. If I fuse them, what do I call this? Protoplasmic fusion, or I will also call this as somatic hybridization. I will also call this as somatic hybridization. Okay, and the things that we are going to get at the end, the thing that I'm going to get, you will call this as the somatic hybrid. This is known as the somatic hybrid. So the plants developed by the fusion of protoplast of two different plants are somatic hybrids, and it is produced with the help of somatic hybridization, where you fuse the protoplast. Remember, it's not soma clones. Do not do that mistake. So you get pomato when you fuse potato and tomato. So that is somatic hybrids. Soma clones are something that you get at the end of tissue culture. You know, when you do tissue culture, when you take a part of the plant and you put it in the nutrient medium, you make it grow, okay, and if at all you are making thousands of them to be grown, you call this as micropropagation. What do you call it? Micropropagation. And when you do that, all the organisms are going to look the same, which is why you call them clones, soma clones. Here they are not looking the same. Okay, nor they are transgenic plants, nor they are GM plants. GM plants as in genetically modified plants. So your only answer for this question would be soma clones. Yes, somatic hybrids. I'm sorry. Okay. Now, identify the incorrect statement. The transgenic cow Rosie produces milk enriched with alpha-lactalbumin. You know, alpha-lactalbumin is a human milk protein, and now we have made the cow produce it. The transgenic cow Rosie is producing this milk which is enriched with alpha-lactalbumin. Is it possible? Yes. That is done using genetic modification. Now, Bt toxin genes CRY1AC controls corn borers. I'll come to this part. Protein, human protein alpha-1 antitrypsin is used to treat emphysema. Emphysema is a respiratory disorder, and is alpha-1 antitrypsin used to treat emphysema? The answer is yes. But how do we produce it? It is produced in the human body. Can we make animals produce it? Yes. So this is also an example of transgenic animals producing our thing. One is the cow Rosie producing alpha-lactalbumin. Here it is alpha-1 antitrypsin for treatment of emphysema. This is important. The commercial production of human insulin was made possible by the transgenic process of the E. coli. Is it? So insulin is our hormone. It is made to be produced by E. coli. Yes, it was done. So again, example of transgenic animals. Now over here, the only point they are left with is Bt toxin genes CRY1AC controls corn borers. Is it correct? It is not. It does not control corn borers, it controls cotton bollworms. So should I tell you the story here itself? I think I can. So listen to me. So I told you Bacillus thuringiensis, which is there, it is a bacteria that produces toxins. Okay, and these toxins are protoxins, as in basically they are inactive. Okay, they are what? They are inactive. Protoxins are there. Now, basically, who gives information to produce this toxins? It is the cry gene. It is the cry gene. And now, when an insect has this particular bacteria, inside the gut, inside the mid-gut basically, this particular enzyme which is there, this particular toxin which is there, it becomes what? Active. The protax toxin becomes what? Active version of toxin, and it leads to perforation of the epithelial layer and later leads to death. So, cry gene is responsible for production of this thing, and the cry genes are very much specific to certain insect groups. So, you have CRY1AC and you have CRY2AB. CRY1AC is there and CRY2AB is there. The next thing that you have is CRY1AB. Okay, CRY1AC, CRY2AB is there. Apart from that, you have CRY1AB. Listen to me. First letter C, cotton. Second letter B, bollworms. So cotton bollworms are being killed with the help of the enzyme, the toxins produced by these two genes. Only one is there, so that is starting with C, that is corn, and ending with B, that is borers. So remember, cotton bollworms is a bigger name, which is why you need two cry genes. One 1AC and one 2AB. Over here you just have one, so that is why 1 is there, and the first letter, last letter is corn borers. So you can remember like that. CRY1AC, CRY2AB. First letter C, second letter B, because it is a bigger term. Over here, first, second, C and B. Both of them are starting with C and B. That is why I am telling you again and again. This is a smaller name, this is a bigger name. So Bt toxin genes CRY1AC controls corn borers. It is wrong. It is controlling cotton bollworms. So this is a wrong statement. Okay? Yes. Now, which of the following statement is correct with respect to synthesis of insulin? Again, now this is a question where you need an explanation. So usually we had the human insulin, but how do we cannot tell our body to produce it in more amount. So we were depending, so people who had diabetes, they were depending on animal pancreas, okay, basically the pigs to produce insulin, the cattle to produce insulin. But when they had this insulin, it was causing them allergies. So then a company came into the picture, and that was Eli Lilly. Okay, Eli Lilly. So Eli Lilly company came into the picture, and they said, why not try to produce human insulin with the help of E. coli? Okay, but there is a problem. The problem is that human insulin is made up of A peptide, B peptide, and joining both of them you have a C peptide. This is found in proinsulin, as in the pro form of hormone. This is a hormone, right? It is found in the pro form of hormone, immature form of hormone. When it is matured, the A and B are the only ones which are there, and the C is removed. The C1 is removed, you just have A and B, and they have disulfide bonds between them. This time you will now call this as a mature form of hormone. So C is being removed within our body with the help of the machineries that we have. And always remember, it's a hormone. It's not an enzyme, it's a hormone. Okay. Now over here, what did Eli Lilly do? It took the gene that is coding for A peptide, and it made one E. coli to produce the A peptide, and it took the gene coding for B peptide and put it in another E. coli. Now this produced B peptide. Now A and B were retrieved, and both of them were joined in the laboratory condition with disulfide bonds. This is how you got the first insulin, and you call it as humulin. You call this as humulin, not first insulin. This is how you got the human-like insulin, which you call as humulin. Okay, so which of the following statement is correct with respect to synthesis of insulin? Synthesized as proenzyme. No, it's a hormone. Synthesized as prohormone and needs to be processed to become what? A functional hormone. To become a functional hormone. This is correct. Synthesized as proenzyme. Wrong. Synthesized as prohormone and needs to be processed to become a functional proenzyme. This is also wrong. So why not, ma'am, put the entire gene over here? Because E. coli does not have machinery to cut the C peptide down. That is why we made sure A peptide and B peptide were put separately. Okay? Yes. Done. Now this is a picture. Please know about this particular picture as well. So you have A, B, and basically this is the C peptide. So A and B were joined with the help of sulfide bonds, and in between you had the C peptide. And then you later removed the C peptide, you just have the A and B in the mature form of insulin. Okay. Crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves, because so bacteria is producing this particular toxin, but it is not killing the bacteria. Why? Because, my dear children, it is inactive. It is inactive. The toxin is inactive. Bacteria resistant to toxin? No. Toxin is immature? No. Toxin is inactive. It does not have the proper environment to become active. It needs an alkaline pH, and it gets it inside the midgut of the insects out there. So bacteria encloses the toxin in a special sac? No, no. Only answer is C. Okay? Yes. Next. Statement 1. The plants genetically identical to the original plants are called soma clones. The plants genetically identical to the original plants are known as soma clones. Is it? Yes. I told you in tissue culture and all you found it. Okay. Yes. So it will be soma clones. Soma clones can be obtained through micropropagation. Can you? Yes. The tissue culture only, whenever you have lot of plants being produced with the help of tissue culture, you call it as micropropagation. So soma clones can be obtained through micropropagation. Yes. Statement 1 and statement 2, both of them would be true. And in tissue culture, always remember, I'm telling you again and again and again, only the apical and axillary meristem part are being taken. You know, meristematic cells are taken. Why? Because they have a potential to divide very, very fast. And they are free of viruses. If at all you want to produce virus-free plant, you take the apical or axillary meristem. Okay? Yes, ma'am. Okay, ma'am. Fine. Now, the pollen grains of a plant with 2n is equals to 48 are cultured by tissue culture to produce a callus. What would be the number of chromosomes in the cells of the callus? What is this, ma'am? So basically, suppose I have a plant. I take a part of this plant. You call this as an X plant. Now it is not a part of the plant, it is X. Okay, that is why X plant. And basically you put it in some media where these cells are going to divide. Now whatever thing is going to be found is known as a callus. This callus is put inside a medium that has rooting hormones such that you have roots being formed. Then shooting hormones such that a shoot is being formed. Then basically you take it and put it in the soil. That's how it goes on. Okay. Now here you are saying the pollen grains. You are taking the pollen grains of a plant. You're not taking the plant, you're taking the pollen grains of a plant. The plant is having diploid nature, 48. The pollen grains will not be diploid, it will be haploid in nature. The pollen grains will be having only 24 number of chromosomes. And now, what would be the number of chromosomes in the cells of the callus? So since you have taken the pollen grains which have 24 number of chromosomes, obviously all the cells of the callus will also have 24 number of chromosomes. So your answer for this question would be 24. So you have to use a bit of understanding of sexual reproduction flowering plants also up here in this particular question. Yes, yes. Next. Statement 1. Bacillus thuringiensis produces insecticidal protein crystals called Bt toxin. Yes, it does. These toxins exist as prohormones before entering into insect gut. It is protoxin, not prohormone. So this is wrong. Statement 1 is true, statement 2 is false. Next, DNA in a clone of cells followed by detection using autoradiography is called. DNA in a clone of cells followed by detection using autoradiography. What do you call this as? What do you call this as? Template, probe, transcript, cistron. DNA in a clone of cells followed by detection using autoradiography is called, basically, a probe is a DNA which the sequences of which we know already, and basically we are tagging it with some radioactive substance. We are tagging it with some radioactive substance. So DNA in a clone of cells followed by detection using autoradiography is known as a probe. Is known as a probe. Okay? Yes. Now, use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as. So basically, suppose you are the ones who usually produce basmati rice. In your country, it is being produced. I am coming from America and I am saying that, basmati rice is mine, I am patenting it. No one else can produce it. What do you call this? Did I inform you guys? No. Did I give you some money? No. So I call this as biopiracy. I call this as biopiracy. Okay, and I'm saying I'm patenting it. Patenting means basically you're not allowed to do anything. You are not allowed to sell, you're not allowed to grow it, things like that. Okay, that is patency. I'm taking the patency of thing. But anyway, these multinational companies, that is me, I'm not paying you. So this is wrong. I cannot do that. So this is known as biopiracy. And this is the exact thing that happened in India. Okay, the basmati rice basically was patented by some US company. Okay, American company. So that is obviously wrong. So that is an example of biopiracy. Now, which of the following statement is not correct? Most Bt toxins are insect group specific. Are they specific? Obviously yes. RNA interference takes place in all eukaryotic as well as prokaryotic organisms as a method of cellular defense. We'll pause this, wait. Recombinant therapeutics do not induce unwanted immunological responses in humans. The recombinant therapeutics, does it induce unwanted immunological response? Obviously no. So this is also correct. Adult onset diabetes can be controlled by taking insulin at regular intervals. So if someone has diabetes means in their blood the level of glucose is way too much, and that can be controlled by taking up insulin at regular time intervals. Yes, it can be controlled. It can be controlled. So this is also correct. So what is left behind is RNA interference, and we are going to see a question related to that. So oh my god, just a second, okay? Okay. So I will talk about RNA interference within a few minutes. Before that, let's solve this question, because here you have everything that you have already studied. Okay, so match column 1 with column 2 and select the correct option from the given codes. Alpha-1 antitrypsin. Why do you need it? Alpha-1 antitrypsin is for treating emphysema. Retroviral vector. Retroviral vector basically is used in gene therapy. Vector is used in gene therapy. So it's not AIDS, okay? AIDS is a retrovirus, but you not make a vector there. So AIDS is caused by a retrovirus, but anyways, you have a better option to match up AIDS. So retroviral vector is gene therapy. D goes with 2. So D goes with 2 and A goes with 3. I have got my answer, but let me see other options. Transposon. Transposon are mobile genetic elements. That means they can move from one area to another. There are segments of DNA that can move from one area to another, causing mutations. ELISA, enzyme-linked immunosorbent assay, which is used for finding AIDS. It is a detection method for what? AIDS. Now with that, I remember one very important thing. If at all, suppose in the question it is being asked, you need to find the small traces of DNA. You need to find the small traces of DNA. What do you use? Which are basically small traces of pathogens. The pathogen is there, but it is in very less amount. What method do you use to amplify it? PCR. PCR is there for amplification of those contents so that you can easily find it out. Okay, remember that. So I know RNA interference is left behind over here. All the other topics were correct. So we will study about this right now. So we are going ahead with the 5 marker questions from this chapter. So my dear children, I'll be going to RNA interference. Where is it? Yeah, here I have RNA interference. So what is RNA interference? How does RNA interference help to develop resistance in tobacco plant for nematode infection? That is the question. So how does it help? Are we ready to find it out? Let's go. So over here, this is the tobacco plant, okay? And the tobacco plant is affected by a nematode, that is Meloidigyne incognita. Now we need to stop this infection somehow. Okay, we need to stop this infection somehow. What can be done? What can be done to stop this infection? It's very simple. Listen to me carefully. This particular organism has its own DNA and it basically produces what? An RNA. It basically produces RNA, complementary to it, right? Now this mRNA has to undergo translation, to produce proteins. mRNA is usually single-stranded. If at all it is made to be double-stranded, it cannot undergo translation, it cannot form the proteins, it cannot cause any trouble up over here. So that thing is being utilized. From Meloidigyne incognita, you take up the genes which is responsible to produce a double-stranded RNA. You take the genes which will be coding for what? Production of RNA. And basically that gene is inserted inside the plant. And that gene will now produce what? The double-stranded RNA. It's there in the plant's body. This double-stranded RNA which is there, it goes to the mRNA of the Meloidigyne incognita. Now it is double-stranded, but it will break the bond and come over here and make a new bond up over here. You get it? I'll tell you again, listen to me carefully. Basically, I put the gene over here in this particular plant with the help of Agrobacterium tumefaciens because that is my vector. Now the plant is producing double-stranded RNA. The plant already has the mRNA of what? This organism. From this double strand, one strand is going to come over here and form a double strand over here. One strand is there, one strand will come over here and make a double strand over here. Do you think it will be able to undergo translation? No. The translation process will be blocked. It is a pre-translational modification. Before translation happens, this has to be done. You get it? Here double-stranded, the RNA is being used, and my dear children, the vector that we use is what? Agrobacterium tumefaciens, because we are dealing with plants. Now, this is a defense mechanism that we have in eukaryotes. Eukaryotes have this defense mechanism. Prokaryotes do not have it. Listen to me. Eukaryotes have it. Eukaryotes as in plants, animals, all of it. Bacteria do not have it. Bacteria is a prokaryote. Okay, and now how do you write the steps? It involves silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA. What do we do? Identify the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant. Then, using Agrobacterium tumefaciens to insert the gene. What do we do? To deliver the silencing gene, we take the help of Agrobacterium tumefaciens. Later, production of double-stranded RNA in the plant. In the plant, they produce double-stranded RNA. Now, RNA interference in the nematode. This double-stranded RNA triggers RNA interference in the nematode, silencing the target gene in the nematode cell. As a result, the nematode can no longer function properly and cannot cause harm to the plant as well. Okay, so this is the thing that you need to remember. Now, if at all we come back to this question and we try to solve this, will we be able to do this? So, which of the following statement is not correct? RNA interference takes place in all eukaryotic as well as prokaryotic organisms. Wrong. It is just eukaryotic organisms. Now, genetically modified plants have been developed through core techniques of biotechnology. How are these genetically modified plants useful to humans? How are they useful to humans? First, basically, when you genetically modify a plant, it has increased mineral usage. So, it can survive even when there is no minerals in the soil. Not much minerals, not much minerals in the soil, still it will be able to survive. Resistance to pest and diseases, you saw the example of Bt cotton and also RNA interference. That is genetically modified plants. Now, reduced use of chemical pesticides. Okay, there is reduced use of chemical pesticide. Why? Because, my dear children, the plants are already pest resistant. So, if chemical pesticides are less, chemicals are less. Now, tolerance to abiotic stress. Genetically modified plants, they can be engineered in a way that they can be resistant to some environmental stresses such as salinity, drought and things like that. Now, improved nutritional quality. Okay, so over here, genes can be added to increase vitamins, minerals or proteins. Genetically modified plants might have other benefits as well. For example, I am putting a gene that is going to give or that is going to produce vitamin A inside what? Inside rice. Inside a variety of rice. Now the rice will be enriched with what? Vitamin A. So the people who are having the rice, they are getting better nutrition quality, and this is exactly what happened. There were malnutrition happening in Africa. At that particular point of time, this particular rice was also given so that at least they have vitamin A and things like that. So to improve the nutritional quality also, we do genetic engineering, and this is also a benefit of genetically modified plants. Example is vitamin A. Okay, next thing, reduces the post-harvest losses. So genetically modified plants, my dear children, they can again survive under moist condition also. They can prevent the growth of fungi and things like that, which is why it will be slowing down the spoilage also. So these are the few points that you need to remember. Next, briefly explain the use of transgenic animals. First use is to study of normal physiology as well as development. Suppose there is an organism out there, and suppose we are there and we have insulin-like growth factors. Okay, insulin-like growth factor is there. We do not know the exact function of this. We put it inside an organism and see what functions do they perform by observing the changes that are going to happen in the growth and the development. Get it? So to study the normal physiology and development, we can use transgenic animals. Second, to disease study and human models. Basically, I want to see what exactly happens in cancer. So when I know what exactly happens, I will be able to treat it better. You get it? So basically, animals were created as models for human diseases such as cancer, cystic fibrosis, arthritis and Alzheimer's. Remember their names, it's important. Cancer, Alzheimer's, cystic fibrosis as well as arthritis. Okay, now for production of biological products, you already saw that alpha-1 antitrypsin is being produced by animals. It is used in emphysema treatment. Apart from that, Rosie the cow is producing what? The nutrient-rich human milk, that is alpha-lactalbumin-rich milk. Now for vaccine safety testing, I made a vaccine. I want to check its safety. I insert it in an organism and see its effect. I want to see the chemical and toxicity testing. For example, how much toxic is this particular chemical? Can it be given as a drug? So what do? I make the organism sensitive and then I give this drug and see what is the effect. If at all the effect is less in a very sensitive organism, that means this particular drug is safe to be given to other human beings as well. So I know this is very bad to say, but yes, this is the uses or applications of transgenic animals. Okay, so this is there in the model question paper also. Make sure you practice them. Now, explain the application of biotechnology in the field of medicine. We have already discussed it. One is insulin, another one is genetic engineering, that is gene therapy. Always remember, gene therapy does not have a, usually we do bone marrow transplantation. You also give enzyme replacement therapy, where you basically give the enzyme transfusion again and again. But all of it needs what? Repeated transfusion. The only permanent cure, if at all, that is there, it is to basically treat it during the embryonic stage itself. To treat it during the embryonic stage itself, otherwise no. Okay, so permanent cure is possible by introducing the ADA gene into the embryonic or the stem cells genes. That's all. Okay. Now, ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? So what is the principle of this technique? Antigen antibody binding is the principle. And name the disease which can be detected by method. It is AIDS. So how does it work? So I'll tell you. Suppose a person has AIDS, but I want to assure it. I want to make sure of it. So I have a well like this, where I already have primary antibodies. I have primary antibodies, and these antibodies I know are specific to antigens of AIDS. My dear children. So I have these antibodies. I take the blood of a person who I assume has AIDS, and that is being treated. If at all that person has AIDS, my dear children, obviously will be having antigens, and that antigen is going to bind specifically on this antibodies, but I cannot say it, which is why I'll need another antibody, secondary antibody. This antibody is linked to certain enzymes. Okay, what does this enzyme do? Basically, when I add a chromosome or coloring substance, this enzyme basically gives another color out there. It changes its color out there, indicating that, hey, yes, the interaction is properly occurring, and basically that this particular person has what? AIDS. So it totally depends on antigen antibody binding. So it works on the basis of antigen antibody interaction. A specific antigen or specific antibody is attached to a plate. The patient's sample is added. If the patient has the matching antibody or antigen, they will bind together. Okay, an enzyme-linked antibody is then added. A coloring agent, chromogen, is also mixed. The enzyme reacts with the chemical to produce what? A colored product. A color change means the antigen or antibody is present. Disease is detected. Okay, no color change means no disease. Name the disease. It is what? AIDS. Next question. Why are meristems preferred in tissue culture? Why are meristematic cells preferred in tissue culture? First, they can divide rapidly. Second, I told you they are virus free, and this is very important. Axillary and apical meristem. Okay, that is, so they are dividing rapidly, and the second thing, they are usually virus free. So this can be asked like an MCQ, which is why I added it. And now you have RNA interference technique. Okay, and we discussed that. Remember, tobacco plant, and it is Meloidigoin incognitia, which is the nematode's name. Now, one of the applications of biotechnology is to get pest-resistant plants. Justify the statement with reference to Bt cotton. So, in Bt cotton, as we have discussed initially, cry genes are there, cry 1AB, cry 2AB. Basically, it is there to kill insects belonging to lepidopterans, coleopterans, dipterans. Please remember lepidopterans, coleopterans, and dipterans. Over here, they produce protein crystals, which are there, and they are inactivated but get activated in the alkaline pH of the gut, and it creates spores in the epithelial cells, killing the whole organism. So just read this, that's all. Rest we have discussed. Now we are going ahead with the next chapter, that is organisms and population. So I see it's almost up to 2 hours out there. So my dear children, I hope you have covered the previous 3 chapters nicely, and now we have 3 more chapters. Hopefully they will be covered in a faster pace. Why? Because they are small. So yes, let's begin. Just give me a second, I'll drink some water, and you also. So finally my bottle is over almost. Okay, let's go. So you will have a one marker question and one 5 marker question coming from this chapter. Let's see. The organisms that produce only once in their lifetime. What do you call these organisms as? They produce only once in their lifetime. You call them as simulparous organisms. Okay. Oysters, pelagic fish, salmon fish, or hippocampus. What is your answer for this question? It is salmon. It is Pacific salmon actually. And also bamboo trees. They also reproduce only once. We have studied this. right? So Pacific salmon or bamboo tree, both of them basically are what? Similparous organism. They breed only once. Heteroparous means they breed multiple times. For example, fishes, mammals, all of them. Okay, done? Yes, ma'am. Next. In a pond, if there were 20 lotus plants last year and through reproduction 16 are added, calculate the birth rate. So we had the initial population to be 20, okay, and through reproduction 16 of them are also added. Calculate the birth rate. So you do like this. It comes out to be 0.8. Okay, it comes out to be 0.8. But now how do you write the birth rate? You have to name the organism and then say the year, okay, because it's rate. So 0.8 offspring per what? Per lotus, right? Per lotus per year. That will be your answer. 0.8 offsprings per lotus per year would be your answer for this particular question. Okay, so number of new individuals added, initial population into the time period which was there. So basically, if here year is being mentioned, so you have to talk about year. If week is being mentioned, you have to say per week. So whatever timeline is given, please go according to that. Next question. The correct order of various levels of biological organization in an ecosystem is, remember this is standing for the arrows. So the correct order for the levels of biological organization, first we have organisms. Organisms comes together to form population. Population comes together to form community. Community forms ecosystem and ecosystem forms biome. This is your answer. The first question, the first option that we have itself is the answer. Okay. This is also a model question paper question. Next, Jiva was growing a bacterial colony. This is previous year model question paper question, okay? Jiva was growing a bacterial colony in a culture flask under ideal laboratory conditions where the resources sooner or later becomes limiting. Which of the following equations will represent the correct growth in this particular case? So over here, my dear children, you have two type of growth. Okay, one growth is basically where it goes exponentially. This happens where the resources are not limited. If resources are limited, you have a carrying capacity. Carrying capacity is a maximum population size that a environment can handle because the resources are limited. In that particular case, what is happening? You see a sigmoid curve. You see a sigmoid curve. This is known as logistic growth. Logistic growth. So over here, bacterial colony is there, but again, it is mentioned that resources are limited. In this particular condition, obviously, it will be logistic growth. Logistic growth that will be considered. Now here you have the formulas of logistic as well as exponential growth. So you have to tell which one is the correct option. What is the formula for logistic growth? dN by dt is equals to Rn. R is not capital, R is small. R is intrinsic rate of natural increase. Okay, until here this is a formula for exponential growth. Now if at all you put carrying capacity also here, it becomes the formula for logistic growth. So dN by dt is equals to Rn, k minus n divided by k. K is carrying capacity. So your answer for this question becomes this. It's not plus, okay, it should be minus. Cool. Yes, ma'am. Done, ma'am. Okay, done. Fine. Next. Monarch butterfly is avoided by the predators because, why is monarch butterfly avoided by the predators? Do you know that? Because they are highly distasteful. So monarch butterfly in the caterpillar stage, they go and feed on, okay, they go and feed on some milkweeds which are very toxic, okay, and which is why, and they store the toxin in their body such that when they become adult and someone have them, it is very distasteful for them. Okay, so monarch butterfly is avoided by the predators because it is cryptically colored? No. It is highly distasteful? Yes. It produces a poisonous cardiac glycoside? No. It has stinging cells on its body? No. Your only answer for this is B. Next, what does the shape of the given age pyramids A to C reflect about the growth status? So A, B, C is given. You have to tell what is happening up here. So this is the pre-reproductive age group, as in the ones that will mature and reproduce later. Then you have reproductive and then you have post-reproductive, that is the aged group out there. So if pre-reproductive is in more amount, that means in future they are going to reproduce and increase the population, yes. So can I say it will be expanding? Yes, ma'am. So this is the option that I have. A is expanding. B, Both of them, reproductive and pre-reproductive, are in the same amount. That means it's a stable environment that is being created. Over here, if you see, the pre-reproductive group is very less. So in future, there are only less organisms to reproduce and maintain the population size. It is declining. It is what? Declining. These are the age pyramids that we have. So your answer for this question becomes C. Okay? Yes, ma'am. Next, on the rocky sea coast of Scotland, the larger and competitively superior barnacle, Belanus, dominates the intertidal area and excludes the smaller barnacle, Thalamus, from that zone. What kind of interaction is being depicted by this example? So in the intertidal region over here, my dear, suppose initially you had Thalamus, few Thalamus, and you had Belanus also. But these Belanus were dominated once, which is why they made sure that these guys are excluded from here, from the intertidal zones. And basically, they can either move up or go somewhere. But anyway, the Belanus, they kept on dominating and they spread around. They utilized the entire thing. What is happening over here? Competition. Not predation, not parasitism. It is what? Competition that is happening over here. And if at all you are asked, what is this process known as? Where basically they are utilizing the whole of the resources now. Once the other organisms are moved away, it is utilizing everything. This is known as competitive release. This is known as competitive release. Okay? Yes. And this was Cornell's experiment. This was Cornell's experiment. So if at all you are asked about which experiment, it is Cornell's experiment. Next question. Assertion. The epiphytes use the trees only for attachment and manufacture their own food by photosynthesis. Have you seen an epiphyte? Epiphyte is something that grows on a mango tree. Okay. So it is said that the epiphyte uses the trees only for attachment and manufactures their own food by photosynthesis. Is that true? Yes. It's just for attachment. They prepare their own food. They just need sunlight, and that is only at the top level. That's why they are there. So they are not harming any plant. Okay, you need to understand they are not harming any plant out there. The reason, commensalism results in negative effects on the growth and survival of one or both of the population. Wrong. Commensalism is happening over here. Okay, the epiphytes are gaining benefit. The mango tree is neither harmed nor benefited. So it's a +0 relationship. +0 relationship. This is commensalism. It's not a negative effect. It does not show a negative effect out there. So what can I say? That option A, assertion is true, but the reason will be false. Assertion is true, but the reason is going to be false. Okay? Yes, ma'am. Next. Abingdon tortoise in Galapagos Island became extinct after goats were introduced on the island. Mention the type of interaction. I have already mentioned it, but let me tell you. So there were goats and there were Abingdon tortoise. Tortoise were the first one which were there, and then goats were introduced. So both of them were feeding on grass basically, and what happened is that when the goats were there, they dominated over this and they made sure that the Abingdon tortoise got extinct. Okay, they didn't give enough resources. These guys got extinct. What do you mean by that? This is known as competitive exclusion, where one organism gets extinct. So this law was put by Gauss. Okay, Gauss said that no two species no two species feeding on the same resources can coexist indefinitely in the same area. One is going to get exist, and that is the example for this. Okay, and this is competitive exclusion. Now we're going to see the long questions of this chapter. Just give me a second. My mic has got stuck somewhere. Yes. Okay. First one. Write an equation to calculate the population density at time t. If at all you are asked to find the population density at time t, how do you find it? Nt is equals to N naught. That means initial population plus birth plus immigration. Birth plus immigration. Immigration means number of organisms coming into the population, minus death plus emigration. The emigration means number of organisms moving out of the population. So always remember, population density has certain factors, and that is births, deaths, immigration and emigration. Births, death, immigration and emigration. Out of that, 2 of them are the most important factors, the major factors affecting the population density. This happens, this is also considered when colonization of new species happens. That is, organisms are moving here and there. Okay. Now, population density fluctuates due to change in 4 basic processes. Mention and define these 4 basic processes. First one is birth rate, natality. The number of individuals added to the population by reproduction per unit time. Mortality, the number of individuals left from the population per unit time. Immigration, the number of individuals entering a population from outside. Emigration, number of individuals leaving a population. So these are the 4 factors. Out of them, I told you 2 of them are super important. Okay. Next, what is mutualism? Mention any 4 examples of mutualism. What is mutualism, my dear children? It's an interaction between 2 species in which both interacting organisms benefit from each other. It's a plus-plus relationship, and you need to know the examples for each of them. Lichen, okay. Okay, before lichen, I'll go with mycorrhiza, since we studied about this. Mycorrhiza is what? Fungus and plant roots. Fungus provides phosphorus and everything, and plants basically provides what? Nutrition. Similarly, lichen is a combination of algae and fungus. So instead of plant, it is algae. Algae provides organic matter, and fungus basically, again, it will be providing food, and not just food, not food, protection actually. Not food, but protection. Protection and very important nutrients are provided by the fungus. So algae and a fungus combination is known as lichen. Apart from that, rhizobium bacteria and leguminous plants. The rhizobium bacteria fixes nitrogen and gives it. Plants gives what? Shelter and also nutrients back to the rhizobium. Fig tree and wasp. Fig tree and wasp. Fig tree basically provides shelter so that the eggs can be laid. Also provides food. Okay, fig tree seeds are being eaten up by wasp. Now wasp help in the pollination. So there are 4 examples that we have. Please remember, lichen is between algae and fungus. L is over here in lichen. You can remember like that. Algae and fungus, lichen. Mycorrhiza is fungus and plant roots. It's plant, not algae. Okay? Yes. Next, name the types of interactions seen in each of the following. Clownfish living among the tentacles of the sea anemone. Sea anemone have tentacles, and in between that clownfish is living. Basically, these tentacles provide safety to the clownfish. Clownfish, does it help the sea anemone? No. So one is getting benefit, other is neither getting benefit nor getting harmed. It is commensalism. It is what? Commensalism. Next thing, koel and cuckoo bird. During the breeding season. Koel and cuckoo bird. Okay. Actually, okay. So crow and cuckoo bird during the breeding season. So in that particular case, what exactly happens is that the cuckoo, okay, the cuckoo which is there, it has developed a way in which it resembles the egg of the crow. So the crow had laid its egg, the cuckoo also comes and lays its egg over here. Remember over here, this parasite which is there, it shows brood parasitism. So basically, why parasitism? Because one organism is basically taking the benefit of the other organism. So over here, it's a plus-minus relationship. But you might be thinking, why a plus-minus relationship? Listen, cuckoo is getting benefit because its egg is also being nurtured. But why is it minus over there? Because crow has to provide its energy into nurturing the cuckoo's egg also, right? So it is a plus-minus relationship out there because it is wasting its energy for some other organism out there. It is known as brood parasitism. What do you call it? Brood parasitism. And always remember, it's not the host egg which have evolved. It's the parasite's egg which have evolved to look like the host egg. So over here, it is brood parasitism. Now, barnacle growing on the back of the whale. Whale is there on the back of the whale. There are small barnacles. Again, barnacles are there on the whale so that they can be transported easily. But whales not get any benefit. So, it's a plus-0 relationship. Again, that is commensalism. Marine fish and copepods. Okay, just like how lice are there on human beings, how ticks are there on animals, copepods are there on marine fish. Copepods are parasites. So, it's a plus-minus relationship. Okay, they are parasites, so parasitism is there. Sexual deceit in Mediterranean orchid Ophrys. So Ophrys, which is there, it's a bee. Oh, sorry, orchid which is there, it is, sorry, how can orchid be a bee? Orchid which is there, it's a plant, and there is a bee. Okay, there is a bee out there. This bee comes and pseudocopulates on the plant. Why? Because the plant has made itself look like the female bee. Okay, it looks like the female bee, which is why it comes in pseudocopulates. What's the benefit over here for the plant? Basically, whatever pollen grains are there on the surface of the bee is now shed down onto the plant. That is the benefit of it. Okay, that will be the benefit. Now, here it is cheating actually, sexual deceit as in cheating. But anyway, both of them somehow are getting benefit of it. Bee, which comes, my dear children, takes up some nectars, pollens and everything out there, which is why it's a plus-plus relationship. Even though it sounds bad, but yes, it's a plus-plus relationship. So it shows mutualism. It shows mutualism. Okay? Yes. So explain the fascinating forms of interaction in brood parasitism and sexual deceit. We already discussed it. Brood parasitism is where a reproductive strategy where one species, the parasite, lays its egg in the nest of another species, the host, leaving the responsibility of raising its young to the host. Examples: cuckoos are well-known brood parasites. Cuckoo eggs form often mimic the appearance of the host eggs, reducing the likelihood of what? Rejection. Impact on host: the host bird invests resources in raising the parasite's young, often at the expense of its own offspring. That is why it is a plus-minus relationship. Now, sexual deceit, the sexual strategy where organisms use deceptive tactics to exploit the behavior of another species or individual for their own benefit. Then you give the example. Certain orchids such as Mediterranean orchid Ophrys mimics the appearance and scent of female insect to attract the male pollinators. Okay, all of it, as I said, is written over here. Now, mention the population interaction exists among the following. I have already written it, but you should know. Abingdon tortoise and goats, competition. I told you goats compete Abingdon tortoise out. Tiger and deer. Tiger and deer is not parasitism. Okay, here it is written as parasitism. It is predation. Predation. Tiger predate on the deer, which is the prey. Sea anemone and the clownfish. Sea anemone and the clownfish. I told you clownfish is between the tentacles of the sea anemone. So clownfish gets the protection, but sea anemone is neither harmed nor benefited, so commensalism. Wasp laying its egg in the fig tree. It is mutualism. Cuscuta growing on the hedge plant. So cuscuta which grows on the hedge plant, it shows parasitism. It shows what? Parasitism. Okay, it says a parasite. Cuscuta does not have chlorophyll of its own, which is why it is dependent on the plant for food. Cool? Yes, ma'am. Fine. Now, what is commensalism? Give any two examples. This is interaction in which one species benefits and the other is neither harmed nor benefited. Orchid growing on an epiphyte, as an epiphyte on a mango branch. Here, orchid gets the benefit of sunlight and everything. Mango is not harmed. Clownfish living among the tentacles of sea anemone. Barnacles on the back of the whale. They are getting easy transportation, that's all. Cattle, egret and the grazing cattle. So when the cattle moves around, there is a small bird that comes and sits. No, it basically, when the cattle moves around, it makes sure that the whatever insects are there in the grass, they move up. And the cattle egret feeds on it. So it's a +0 relationship again. Now state Gauss's competitive exclusion principle. It says that two closely related species competing for the same resources cannot indefinitely and cannot exist indefinitely, and basically the competitively inferior one will be eliminated eventually if the resources are limited. Okay? Yes, ma'am. Now, what are ectoparasites and endoparasites? List any 3 parasitic adaptation. Ecto means they are on the surface. For example, ticks, lice and everything. Endo means they are inside the body, such as tapeworm and everything out there. Okay, now list any 3 parasitic adaptation. What are the adaptations that parasites have? Presence of adhesive organ suckers to cling to the host. Remember, they do not have hand, legs and all. So basically, they need suckers to cling to the host. Second thing, they do not have unnecessary sense organs, eyes, nose, teeth, they don't need this. So loss of unnecessary sense organs, because that will be not allowing them to move easily. Okay, no protrusion should be there in the body. Their body should be like this, slime. Now, loss of digestive system, because my dear children, they are already feeding on the simpler nutrients out there in our body. So they do not need a digestive system for their own. High reproductive capacity, so that they can increase the number and affect us more. And complex life cycle involving 1 or 2 intermediate hosts. Vectors to facilitate parasitization in its primary host. My dear children, in your book and in your NCERT book, example is given of human liver fluke. Please understand and please remember, you need 2 hosts in the liver fluke. One is a snail and other one is a fish. There are 2 intermediate hosts in the liver fluke. One is a snail and another one is a fish. Okay? It's an endoparasite. Now, predators are prudent and conduits in nature. Support your answer by giving the reasons. So, prudent basically means that an organism makes sure that it has anything to eat every time. it does not overexploit its prey. For example, I am there, I cannot go eat everything out there. I need to make sure, if at all my resources are limited, I need to make sure I leave behind something so that I have food later on also. So predators are prudent because basically they do not overexploit the prey, because they know that the prey, if it is exploited, they will go extinct, and in that case they will also, the predators will also go extinct. That is why we say they are prudent. So they are considered prudent because overexploitation of prey would lead to their own decline due to food scarcity. Now, why do I call that they are conduits in nature? Because predators, when they, conduits means channels of flow. So an organism, rabbit is there, it is a predator for what? For grass out there, right? So whatever energy stored in the grass is now transferred to the rabbit. Now a tiger comes and eats the rabbit. In that particular case, again, the tiger is act like a conduit of energy, a channel for energy. It keeps on going. So predators act as conduits in the energy flow of an ecosystem, transferring energy from prey species to themselves and the other trophic levels as well. Okay? Yes. Now to lessen the impact of predation, prey species have evolved different defensive mechanisms in plants and animals. Support your answer by giving one example each for plant and an animal. So prey species have evolved some defense mechanism, is it? Yes. So we should know one examples for both plants and animals. In animals, such as insect and frog, they are cryptically colored. This is a defense mechanism that they have. As in, they camouflage. For example, I color myself black and I come over here and stand near the black thing. Will I be visible? No, because I am showing camouflaging. And with that, no one can spot me easily. Now, some are poisonous and therefore avoided by the predators. Okay, some organisms, they are poisonous, which is why no other organism will be coming and eating it. For example, monarch butterfly. Monarch butterfly, my dear children, this is highly distasteful, which is why no organism will prefer to come and eat it. So please remember two examples. One is insect and frog showing camouflage. Second one is monarch butterfly, which is highly distasteful. Now in the case of plants, you have multiple things. First, morphological defense. That means basically I have a morphology having thorns and everything. Now no organism can capture me easily because these thorns will prick. This is seen in the case of acacia as well as cactus. Acacia and cactus have morphological means for defense. Now there are chemical means for defense as well. There are chemical means for defense, for example, calotropis. Okay, calotropis, which is this plant, my dear children, it has some cardiac glycosides. This is important point. It has some cardiac glycosides which affect the organisms which tries to eat it. It has cardiac glycosides which affects the herbivores which will be trying to eat it. Always remember it. It is calotropis. It is calotropis. So that is what a chemical way of defense. Apart from that, always remember there are other chemicals also being produced which we use for our own good. That is nicotine. It's a drug. Quinine. Okay, quinine, nicotine, all of it is used by human beings for their own purposes as well. But they are actually the natural chemical defenses found in the case of plants. Okay? Yes, ma'am. Now, write any 4 factors that influence the population density. We know natality, mortality, immigration, emigration. Now, draw a graph showing exponential growth. This is the graph. Population density on this side, time on this side, and it grows upwards. It grows upwards. dN by dt is equals to Rn, where R stands for what? R stands for intrinsic rate of natural increase. Always remember, R means birth minus death. Okay, and it stands for intrinsic rate of natural increase. And according to Darwin, R is basically reproductive fitness. Reproductive fitness. If an organism is reproductive fit, basically it increases the population size. If high R value is there, that means basically the population size is gonna increase. So this is population density by time, and this is the exponential growth curve. It is a J-shaped curve. In logistic, you get a S-shaped curve. That is why logistic S is there. Okay. Why is this question repeated? Just a second. Okay. Done. So this is it for as of now from this particular chapter. If anything other thing important comes up, definitely let you know. But yes, as for now, these are the things that you should definitely on priority complete. Okay, from the chapter organisms and population. And now we are going to go ahead with the next chapter, that is ecosystem. Again, you have a 1-marker question and 1 3-marker question coming from this chapter. So you please pause the video and drink some water. I will be going ahead because as of now my water is almost done. Yes, I do not have any water left, or little is left. Yes, it's finished. Okay, so listen to me carefully. The decomposers especially abundant in the bottom of the pond are. So usual decomposers that we have, my dear children, they are fungi and they are bacteria. They are usually fungi and bacteria. I call it Facebook. But in the case of pond, there are one more type of decomposers, and that are flagellates. So in the pond ecosystem, you have flagellates, fungi and bacteria. This came in this year's MQP1. Okay? Next, the correct sequence of steps in the process of decomposition is fragmentation, leaching, catabolism, humidification, mineralization. What is the first correct step over here? So I would love to explain you this particular thing with the help of a direct answer itself. Just give me a second. So over here I have the question. Listen to me. How does decomposition work? Okay, listen to me very, very carefully. My dear children, we have complex organic matter. Okay, they are what? They are complex. We need to break it down into simpler inorganic matter. Okay, how do we do it? How do we do it? We have to have some process, and this process is known as decomposition. Listen to me carefully. The first process is to break the larger substances down to smaller fragments. So, when I am breaking the larger things down to smaller fragments out there, I will call this process as fragmentation. And this is usually done by not decomposers but detritivores. Detritivores as in you have your earthworm and everything out there. So they eat the things, they poop it out. Okay, when they poop it out, basically it's the smaller particles that they're pooping it out. Now you have the smaller things out there. Over here, while the breakdown is happening, some inorganic nutrients you might have got. Okay, while the organic matter is being broken down to smaller things, some inorganic matter will be there. It will be now quickly going into the soil. Some water-soluble nutrients will be moving into the soil, and this particular process which is there, you call this process as leaching. This is the second step. The third step, whatever is left behind, we have to break it down to form the inorganic matter. This is known as catabolism. And who does it? It is the decomposers. Decomposers as what, my dear children? Fungi and bacteria. Do you think it's a one-step process? No, ma'am. So over here, while this breakdown is happening, you reach a partially digested stage. You call this stage as the formation of humus. Or you call this process as humification. Here is where the organic matter is partially digested now. Not fully, but partially digested. So this will be the fourth step. So this comes under catabolism itself, but yes, in your book it is put separately, which is why I am telling you separately. Over here it is partially digested form. It is highly nutritious because it is partially digested. It keeps on giving chemicals, nutrient down there into the soil. It is dark, it is amorphous substances that is being formed over here. You call it as humus. Now finally it will complete its breakdown, and you call this process as what? Mineralization. You will be calling this process as what? Mineralization. So towards the end, basically, you have finally broken things down into inorganic nutrients, and this is the fifth step. So 1, fragmentation, 2nd, leaching, 3rd, catabolism, 4th, humidification, and the 5th one is what? Mineralization. Remember, decomposers which are there, they need 2 things. One is oxygen. The second thing that they need is what? Warm and moist environment. They will not be able to do this process when oxygen is not there, that is anaerobic condition, or in colder environment, which is why in the sea, in the water body, in aquatic bodies, the detritus, the breakdown of decomposition process is very much slow. It will be very much slow. Okay, so oxygen, warm and moist environment is needed. If at all the detritus, the dead and decaying matter is rich in lignin or chitin, if at all it is rich in lignin or chitin, the whole process slows down. And if at all it is rich in nitrogen as well as some sugars, the process speed increases. Okay? Yes, ma'am. So that is a question that I have over here in the form of a 3-marker that has also come in the model question paper is what I believe. But over here, let's see this question now. Okay. So the correct sequence of steps in the process of decomposition is what? Fragmentation, then leaching, then catabolism, humidification, and then mineralization. This is your answer for the question. Okay? Yes, ma'am. Next question. The rate of formation of new organic matter by producer. A producer is producing organic matter. What do you call this as? The rate of, if at all you want to say, the rate of production of the organic matter by producers is what? Net primary productivity, secondary productivity, gross primary productivity. Primary productivity. We do not call it gross primary productivity. Gross primary productivity is a total of all the organisms' production. Okay, all the producers coming together and they are producing it, you call it gross primary productivity. Here we are talking about rate of formation of new organic matter by a producer is known as what? The primary productivity. Okay? Yes, ma'am. Now, which of the following food chain is the major conduit for energy flow in terrestrial and aquatic ecosystem? So we have two types of food chain. One is grazing food chain, another one is detritus food chain. Detritus means from the dead and decaying matter, the food chain starts off. So dead and decaying matter is broken down by decomposers and the things are being passed to the soil, and from the soil plants comes and things like that. That is detritus food chain. Do you think it will be more prominent in aquatic ecosystem? I just told you decomposers do not work really well in aquatic ecosystem. Why? Because in the lower environment it is very much cold. So obviously in aquatic ecosystem, it has to be grazing food chain. Grazing means producers as in plants are there, and feeding on the plants you have some other organisms out there. So grazing food chain is something that is prominent out there, whereas in terrestrial, my dear children, it is detritus. It is detritus. So over here, the answer for this question becomes C. This is important question. Okay, it's an important question. Next. If 10 joules of energy is available at the producer level, then the amount of energy present at the level of secondary consumers is. So you have a producer out here, that is the plant. Now the plant is eaten up by a rabbit. This is the primary consumer, right? Secondary consumer is the next organism, suppose it's a tiger. You are being asked, if 10 joules of energy is there at the producer level, how much energy will be there present at the level of secondary consumer? So you should be knowing only 10% of energy is transferred from one organism to another. This is one trophic level to another. So 10% of 10 is equals to what? 1 joule is over here. 10% of 1 will be what? 0.1 joule. 0.1 joule will be your answer. Remember, it's 10% law. What about the rest of the energy? It is lost in the form of heat. It is used up by the organisms themselves. They also have to survive, right? So that is the thing. Okay, this is 10% law, which is why the energy of pyramid is also always going to be upright. It is never going to be inverted. It is always going to be upright because it follows the 10% law. The maximum amount of energy is gotten by the plants. How much? No, sun produces almost 50% of the sunlight can be used by the plant, but the plant uses only 2 to 10% of it. Only 2 to 10% of it is utilized by plant. Only 2 to 10. Okay, so it is the first one to capture it. So obviously it will have highest amount of energy. Then comes the other organisms out there. Now, so please remember this data also. 2 to 10% or 50%. This is photosynthetically active radiation. Out of that, only 2 to 10% is being taken by the plants. Now, given below is a table of factors affecting the microbial decomposition, means favoring, and minus means inhibiting, lowering. Select the correct option. High temperature, lack of oxygen, moist environment, lignin and chitin, and detritus. Okay, tell me the answer. High temperature, does it favor decomposition? Yes, it does. So plus would be correct. This is going to be wrong. Next, lack of oxygen. Is it good? Is it plus or minus? Lack of oxygen is what? Should be there, should not be there. Should be there, should not be there. Should be there. Tell me. Should not be there, right? It should be having oxygen. Then only things will have happening. Moist environment should be there. Yes. Lignin and chitin and detritus should not be there. In the detritus, if at all it's there, my dear children, the whole process will go slow. So your only answer for this question, wait. What did I do? Did I do something wrong? Means favoring, minus means inhibiting. Plus, okay, high temperature is plus. Lack of oxygen is minus. Okay, so answer is wrong here. Okay, the answer is here. The one that I gave you is the correct answer itself. Okay, lack of oxygen is an inhibiting effect. So the answer for this question would be B itself. Now, what type of ecological pyramid would be obtained with the following data? Secondary consumer 120 gram, primary consumer 60 gram, primary producer 10 gram. So this is a pyramid of biomass because gram is being mentioned over here. So primary consumer is 10 gram only. And then primary producer is 10 gram, primary consumer is 60, and then over here secondary consumer is 120. This is inverted or upright? This is inverted pyramid. This is what? Inverted pyramid. And you know what? Inverted pyramid of biomass is actually seen in the case of aquatic ecosystem. In the case of aquatic ecosystem, you can actually see inverted pyramid out there. How does it work? Do you know that? Do you know how does it work over here? Listen to me carefully. In aquatic ecosystem, my dear children, you have phytoplankton as the producers. Phytoplanktons are they have very less biomass, and feeding on them you have zooplankton which have more biomass. Feeding on them you have smaller fishes which obviously has more biomass, and then you have larger fishes which has even more biomass. So this would be inverted pyramid of biomass, just like this. So inverted pyramid of biomass is the answer. It's not number, it's just biomass because gram is being mentioned over here. Okay? Yes, ma'am. Next, assertion. Some aquatic ecosystems have inverted biomass pyramid. I just told you right now. Yes. The pyramid of energy is also inverted in such case. Pyramid of energy can never be inverted. Can never be inverted. So this is wrong. Always remember, the pyramid of number can be inverted. For example, you have just one tree, and on this one tree you have multiple consumers out there. So pyramid of number, one tree is there. Producer is less, but the consumer is more. That can be inverted. Okay. So pyramid of number, pyramid of biomass, they can be inverted according to the examples, but not always. Okay. But anyway, energy can never be inverted. It will be always upright. Cool. So assertion is true, but the reason is definitely false. Next, we are going to solve some long questions of the chapter ecosystem. So describe the steps of decomposition that operates. We already discussed it. Please know all the features, the conditions. Okay. Now schematically represent the pyramid of energy with appropriate units. So you have to make your own pyramid. So what do? You mention the trophic level 1, trophic level 2, trophic level 3, and mention primary producer, primary consumer, secondary consumer, this. Name the organism, plant, rabbit, tiger, and vulture. And then explain all of this. The 10% law, talk about that. It says that only 10% of energy is available at one trophic level is transferred to the next trophic level. So talk about that, make this diagram, then only you will get 3 marks, and mention the trophic levels along with the units over here. The energy has to be mentioned. Okay? Yes, ma'am. Cool? Just give me a second, okay? Okay. Next, ecological pyramids have limitation. Justify with 3 reasons. The pyramids that we saw, do they have limitations? Yes. Why? What limitations do they have? Do they consider decomposers out there? No. So that is one very big problem, because decomposers are playing a crucial role in the environment. Okay, next thing. They assume a simple food chain, whereas most ecosystem has a complex food web. Yes. Do we have a normal food chain? Tiger will not eat any organism apart from rabbit. No, it will eat, right? So there is a complex food chain, but a complex food web, but over here only simple food chain is being considered. Now they cannot represent organisms that can occupy more than one trophic level. They cannot represent organisms that occupy more than one trophic level. Yes, that is also true. For example, human beings, we can eat plants. That means we are primary consumers. Or we can eat animals. That means we are secondary consumers. But are we referred in both the trophic levels? No. At one time, only one trophic level is being considered. So that is also wrong. So these are the limitations that we have with ecological pyramids. Next, pyramid of energy is always upright, can never be inverted. Give reasons. Why? Energy decreases at higher trophic levels, my dear children. Energy is transferred from producers to consumers with each step resulting in what? Energy loss. So organism is using it, organism is also spending away the energy for its own use, and it is while transfer it is not 100% efficient as well. You get it? So obviously energy decreases at higher trophic levels, which is why it is always upright. Now inefficient energy transfer. Second law of thermodynamics says when energy transfer is going to occur, it is not going to be 100% efficient. That is another thing. Decreasing biomass in organism. At higher trophic levels, there are only fewer organisms compared to lower levels. Okay. Why? Decreasing biomass in organisms. Listen, at higher trophic levels, there are only few organisms such as apex compared to lower levels. This also reflects the decrease in energy. Why do we see very less number of tigers out there? Because obviously only those can survive with a little amount of energy that is being transferred. So it shows that there is no much amount of energy that is being transferred to the apex predators out there. Now, ecosystem stability. An upright pyramid supports ecological balance. An inverted pyramid would destabilize the whole system. So at least write the 3 points, that would be more than enough. Okay? Yes. Now mention the functional components of ecosystem. Yes. What are the functional components of ecosystem, my dear children? Productivity, decomposition, energy flow, and nutrient cycling. So initially it was there, but now this is removed from your syllabus, but please know about it. Productivity, okay, production should be there, otherwise how will anything be maintained? Decomposition, energy flow, and nutrient cycling. Now, what is standing crop? That is also being asked. Standing crop refers to the amount of living biomass or number of organisms present per unit area per unit time in an ecosystem. What is it? The total biomass, okay, amount of living biomass of number of organisms living in a particular area at a particular time. Mostly dry weight is considered. Okay, mostly dry weight is considered, as in water is being removed and dry weight is being considered. Okay? Yes. Now, before I go to the next chapter, since I remember it right now, I should be telling you this. No, nothing. It's in biodiversity and conservation, I'm sorry. So, these are the things that you need to importantly study from the chapter ecosystem. Okay, please do not forget, they are the functional components of ecosystem. Cool? Yes. Now we go to the next chapter, which is biodiversity and conservation. Here you are going to get 2 1-marker questions and 1 2-marker question. Okay, so let's see the questions. The IUCN Red List 2004 documented the extinction of species in the last 500 years includes what? The IUCN Red List documented the extinction of species in the last 500 years include what? How many species were there? 784. 784, my dear children. Okay. In that, 338 of them were vertebrates, 359 were invertebrates, 87 were basically plants. 338 vertebrates, 359 invertebrates, 87 plants. This can also be a question. Okay, so if at all you have seen the pie chart, you know vertebrates, invertebrates and plants are given in the same order, you can remember. Okay, so initially invertebrates are given, but over here, yes, you can change the order anyways. So in that case, invertebrates in the highest, and then vertebrates, and then you have plants. Extinction has happened for all these organisms. Okay, now, the following are the in situ conservation approaches except. In situ means you're conserving the organism in their natural habitat. So you're asking what is an exception out there. Natural parks, wildlife sanctuaries, biosphere reserves, all of them are in situ conservation method. What about botanical gardens? No, that is an ex situ conservation method. You're conserving outside the actual habitat. Okay, so that is botanical gardens, as in you get plants outside their natural habitat. So this is an exception. Next. Alexander van Humboldt observed that within a region, species dash increased with increasing explored area, but up to a certain limit. For example, he said that, so over here you have 2 options, richness and you have diversity. So Alexander van Humboldt observed that within a region, species diversity increased within increasing explored area, but up to a certain limit, or is it richness? It is richness. It is richness. And basically, listen to me very carefully. He said, in simple words, suppose this is the area that I am having. The number of species over here will be very limited. If I go around, if I move out, obviously the number of species I will be seeing will be more. He said it is going to keep on increasing, but only up to a certain limit. Beyond that, it is not going to go. And he basically put down this particular thing. So over here you have species richness. and over here you have the area. So more area, more species richness, right? But over here he got a rectangular hyperbola saying that yeah, it is going to be stable somehow, somewhere. It is not going to be going to the upper limit anyhow. And this basically is said S is equals to CAC. But how do you come with the conclusion like that? Listen to me. We say more species richness is proportional to the area that is approached. Is it true? No, ma'am. There is a limitation. Okay, there is a limitation only up to a certain limit. So how do you express that? You basically take it into proportionality constant. That is C. C represents the proportionality constant, and basically that is the intercept of y. Okay, it is the intercept of y. A, which is there, it will also have Z value. Why? It says that species richness, okay, is going to increase up to this much area explored. So you are keeping a limit out there. So that is what you mean by S is equals to CAZ. Now, if at all you make the logarithmic equation of this particular thing, it comes out to be log S is equals to log C first plus log A plus what? Log A. Here you have Z, you put the Z value in front. You put the Z value in front. That is how you make the log thing. And there will be a slope down over here, this slope. Okay, this slope is there for the log value expression. Now over here, Z which is there, it represents the slope of regression. It represents the slope of regression. What do you exactly mean by that? So basically, the slope, suppose it is, I'm making it over here. The slope, suppose it is like this. And there is another slope which is like this. What do you understand by that? Here the slope value is less, Z's value is going to be less. That means it is going to be around 0.1 to 0.2. That means it is less. In this case, with the amount of area that I am exploring, which is way too much, the species richness is not too much. That means it is not following the principle that we studied. Okay, there is only a moderate relationship between the species richness and the area according to lesser Z value. If the Z value is more, that is, if I say it is from 0.6 to 1.2, 0.6 to 1.2, please remember these values. It means that basically more area and more species richness. Here the species area relationship is being followed nicely. That is why the Z value is more. So wherever, if at all I take a smaller area, there it will be less. If I take the larger area, obviously the Z value will be more. Okay, if I go to tropical forest and see their Z value, here it will be more for frugivorous plants as well as mammals staying up there. You get it? And more, by more I mean it should be coming between 0.6 to 1.2. Okay, got it? So here it is species richness that is being increased, and I explained to you the Alexander van Humboldt thingy as well. So please go through it. This is this, this is this. Okay, and C is the intercept of Y. Okay. Now, the genetic diversity shown by raw Wolfia vomitoria growing in Himalayan ranges produces an active chemical called what? Reserpine. It produces what? Reserpine, my dear children, raw Wolfia vomitoria. This reserpine is important for decreasing your blood pressure. Okay, it reduces the blood pressure as well. As per the species inventory, 70% of the species recorded are. So, you know, most of these organisms which we see, okay, 70% of the species, the highest number of organisms are not plants, not fungi, not bryophytes, it is animals. Now, in the animals, 70% of them are insects. In the animals, 70% of this are insects. Okay, always remember that. And one more very important thing, when you talk about fungi, okay, fungi species is a total sum of fishes, amphibians, reptiles and mammals. Okay, fungi will be more than fishes, amphibians, reptiles and mammals. So out of animals, in animals, my dear children, is obviously 70% of species are animals. In animals, the most of it is insects. And if at all you talk about fungi, it is sum total of all these organisms out there. This is three things that is given in NCERT, which is why I just explained to you. Okay, just have that in your head. Now, in general, species diversity dash as we move away from the equator towards the poles. So when we move from the equator towards the poles, the species diversity is going to increase or decrease. Equator to poles. Pole is more colder, and colder means there is less stability in the environment. Glaciation will happen. If there is no stability in the environment, organism will it be able to survive up there? No. So species diversity is going to be increasing, it will be decreasing. It decreases. Okay, it decreases out there. Cool. Next, the graph given below shows species area relationship. Which of the following equation correctly represents the curve? Which of the following correctly represents the curve? Rectangular hyperbola. S is equals to CAZ. S is equals to CAZ. If at all this line was there, it will be this. Got it? Yes, ma'am. The given pie diagram represents the proportionate number of species of major taxa of vertebrates. Identify the groups A and B. So I'll give you an easy way to remember it. Always remember, fishes came first, right? Fishes came first. From fishes we got amphibians. From amphibians we got reptiles. From reptiles we got birds as well as mammals. We got birds as well as mammals. Birds are in more amount than mammals. Remember that according to your NCERT. But fishes are the most in the invertebrates. So farm and then B is there in between. Okay, in between farm, here birds will also be there. So go in the order and you will be able to remember it. So what is A over here? A is definitely fish. And what is B over here? B is definitely birds. Okay? Yes, ma'am. Fine. So this is the picture that is given. Okay, birds is more amount. After fishes, you have birds in more amount. So, yeah. Now select the incorrectly matched pair. Tannins, narrowly utilitarian. Endemism. Okay, first of all, there are multiple reasons why we should be safeguarding our nature. Okay, and that is, there are multiple. One is narrowly utilitarian reason. It says that we should be saving the nature, biodiversity, because it gives us products. Products as in tannin, raisins, gum, everything. Food, water, everything. Not water, food and everything out there. That is why we should be safeguarding the nature. That is narrowly utilitarian point of view, where you're talking just about the products. Broadly utilitarian view is there, where we say that we conserve nature. Why? Because it gives ecological services such as oxygen, such as pollination is happening, seed is being formed, we are able to eat the fruit, on things like that. You get it? So that is ecosystem services, pollination, all the services, normal thing, pollination, oxygen, and aesthetic view and everything. That comes under broadly utilitarian. And the last one is ethical reasons why you should consume. That is, basically we have a moral responsibility towards conserving the nature. So over here it is said that tannins comes under narrowly utilitarian reasons. Is it? Yes. Narrowly utilitarian reason says that we should conserve the nature because it gives products, and tannin is a product. So this is correct. Next, endemism, species confined to a particular region and not found elsewhere, that is known as endemism. Yes, that is known as endemism, and you call these species as endemic species. What do you call them? Endemic species. Wherever these species are found in more amount, you call them biodiversity hotspots. And there are 34 hotspots in the whole world. Out of that, 3 of them are in India. One is your Western Ghats, one is Indo-Burma, and the other one is Himalayas. So 3 of them in India out of 34 in the whole world. Now, lungs of the plant is Amazon rainforest. Yes, Rajasthan is the sacred grove. Rajasthan, sacred groves that are the last refugees for a large number of rare and threatened plants. No. Listen. First of all, what do you mean by sacred groves? This is it's a part of in situ conservation. Basically, suppose I have an area and I have what do you call religious attachment to that area, and I'm safeguarding that area because of religion reason. But when I'm safeguarding the region, I am also safeguarding all the organisms that are staying up there. This is known as sacred groves. Now, sacred groves are found in many places. For example, there are Khasi and Jaintia hills in Meghalaya. There is Aravali hills in Rajasthan. There is Chanda, Bastar, Sarguja in Madhya Pradesh. Okay, there is Western Ghats in Karnataka as well as Maharashtra. Okay, they are all sacred groves. But at one place, these sacred groves are actually the last way I can save the rare and threatened plants. And that is not Rajasthan, that is actually Meghalaya. That is actually Meghalaya. So Khasi and Jaintia hills in Meghalaya are the ones. So always remember that. This is a question that should not be missed. Cool? Cool. Next. All of the following are included in XO2 conservation except wildlife safari parks, sacred groves, botanical garden, seed banks. Sacred groves are not considered in XO2, it's in situ. I just told about that. Wildlife sanctuary is different from wildlife safari parks. Remember, it comes under XO2 only. Botanical gardens also XO2 and seed banks also XO2. Fine. Next. Assertion, the variation in Rauwolfia growing in different Himalayan regions might be in terms of potency and concentration of the active chemical that the plant produces. So variation of Rauwolfia which is there, there's a plant species, there are different varieties of that in the Himalayan region, my dear children, and the variation might be because of the concentration of active chemical reservoir that is being produced. Say yeah, it's correct. A single species might show high diversity at the genetic level over its distributional range. That is example for this. So at genetic level, my dear children, at the same here, at the location, my dear children, at the genetic level, they are diverse, which is why you have diverse production being, for example, rice, you have multiple varieties of rice, basmati rice, this rice, that rice, that rice, everything, right? So this is genetic, a single species is there, different varieties is there. Similarly, talking about Rauwolfia, you have different varieties of Rauwolfia producing different concentration of active chemical. This shows what? Genetic diversity. This is what? Genetic diversity. So a single species might show high diversity at genetic level over its distributional range. This is correct explanation for this. So both assertion and reason are true, and the reason is the correct explanation of assertion. I know it's kind of tricky, but please remember it. You have more example. One is 50,000 varieties of rice is there, and 1,000 varieties of mango is there, genetic varieties. So they show genetic diversity. Apart from this, these are the other examples out there. 50,000 rice and 1,000, what do you call, mango. Assertion, speciation is a function of time, and tropical regions had got a long evolutionary time for species diversification as compared to temperate regions. So speciation or the formation of species, it is a time-taking process, it is being said. And tropical regions have got this time. Why? Because it is very much stable compared to what? Temperate region. Is that so? Yes. Temperate region, as I said, frequent glaciation, this and that is there. That is why it is not stable. So this statement is correct. Now, reason, temperate regions have undergone frequent glaciation in the past, whereas tropical regions have remained relatively undisturbed for millions of years, which is why tropical areas will have more speciation. Correct. So assertion and reason both are true, and the reason is the correct explanation of assertion as well. Okay? Yes, ma'am. Next, we are going to go ahead with the 2-marker question of this chapter. Please pay attention, and with this, we are going to end the session. First one, differentiate between ex situ and in situ conservation. What is in situ conservation? Conservation of species in their natural habitat. Ex situ means you're conserving outside the natural habitat. In situ protects the entire ecosystem. Ex situ basically protects the selected species only. You cannot protect entire thing. You are going to protect only the selected species. Example, here wildlife sanctuaries, national parks, everything. Here the examples are zoos, botanical gardens, seed banks, and everything are there. Okay, so there are differences between ex situ and in situ conservation, and these are the differences. In ex situ conservation, please remember there are some advanced ex situ ways also. What are those advanced ways? Listen, cryopreservation. Preservation, where basically you are preserving the gametes and all. Seed banks. Okay, we are preserving the seeds out there. Apart from that, you have IVF, in vitro fertilization, that also comes under ex situ conservation advanced method. Now one more is there, I am not able to recollect it at all. So cryopreservation, seed bank, IVF, and one more. If at all you are able to recollect, put it down in the comment section or go check it out in your NCERT because it's super important. What was that? Okay, so there is one more advanced way of ex situ conservation, and that is there. Rest everything is being put down over here. Please know this. Okay? Yes. Yes, tissue culture. I got it. It is what? Tissue culture. Okay, that is also a way of ex situ conservation. Wildlife safari is different. Wildlife sanctuaries are different. Okay, now mention the evil quartet of biodiversity losses in the given habitat. So there are 4 reasons why biodiversity losses is happening. One is habitat loss and fragmentation, where basically we are not allowing organisms to stay upright. Okay, we are going up to the area and deforesting it and doing fragmentation and things like that. Next is overexploitation. We are killing organisms. The best example for this is extinction of stellar sea cow of Russia, and also, my dear children, passenger pigeon. Both of them got extinct because of over exploitation. Then we have alien species invasion, where we bring organisms from other areas, and what happens, it causes trouble to the indigenous species, the local species out there. For example, Nile perch is a fish, very big fish, which was introduced into the Lake Victoria, my dear children, and the native cichlid fishes, the small fishes which were there, they got extinct. They got extinct because of this Nile perch. Then we have Parthenium, that is called grass, carrot grass, Lantana, we have Ecornia, all of these plants, they are plants which are invasive species. Then we have one more example of African catfish, that is Clarius geripinus. Okay, that also killed the native fishes out there, made them extinct. So that is alien species invasion. Now we have co-extinction. When one organism which is dependent on another organism, one gets extinct, other organism also get extinct. For example, you have this fig tree and wasp. They have a relationship, mutualist relationship, where both of them survive with the help of each other. One is lost, the other one will be always automatically lost up there. Okay? Yes. Now write any two advanced techniques used in ex situ conservation. I told you. They are asking nowadays. So any two advanced techniques used in ex situ conservation is cryopreservation, preservation of gametes, embryos, seeds or tissues at a very low temperature, -196 in liquid nitrogen, tissue culture or micropropagation as well, growing plant cells, tissues or organs in a nutrient medium under controlled laboratory conditions. So we are doing it because we are sure that those plants might die later on, so we are culturing them separately. Okay? Now define biodiversity, mention the two different types of biodiversity. Biodiversity refers to the variety of life forms on earth, okay, and there are different types of biodiversity. One is genetic, variations in the genes within the same species. Within the same species, the variation in the gene bringing down things is known as genetic diversity. Example is Rauwolfia vomitoria, showing variation in the potency and concentration of rhizopine. Now, India has 50,000 genetically different rice strains and also 1,000 mango varieties. These are what? Genetic diversity. Variations of gene within the same species. Now, species diversity. Diversity at the species level in a given area. That means I am saying that Western Ghats have more amphibian species than Eastern Ghats. You get it? So, at the species level, it's diverse. Western Ghats have more species of amphibians than what? Eastern Ghats. You get it? So this is an example. Now ecological diversity, diversity at the ecosystem level. I say India has mangroves, desert, forest, this and that, lot of things in comparison to a Scandinavian country like Norway. So we are ecologically more diverse than a Scandinavian country like Norway. You get it? So these are the three diversity types. Please know all of it. Please know their examples as well. Now, what do you mean by endemism? Endemism, I said, the species confined to a specific region. Okay, they are known as endemic species, and this process is known as endemism. Now, mention any two causes for biodiversity losses. One is habitat loss and fragmentation, other one can be overexploitation. Talk about the examples and that's done. Okay, but please know all the examples of alien species invasion, super important. Okay, here Steller sea cow and passenger pigeon is an example. Here fig and wasp can be an example. Now give any two examples of sacred groves in India, super important, and I told you in Meghalaya, Khasi and Janshia hills are the last refuges for protection of rare and threatened species. So Khasi and Janshia hills of Meghalaya, Aravalli hills of Rajasthan, western Ghats of Karnataka as well as Maharashtra, okay, as well as Maharashtra, and Sarguja, Chanda and Bastar region of Madhya Pradesh. There are many reasons for conserving biodiversity. Briefly explain the reason for conserving biodiversity from narrowly utilitarian point of view. Why? Narrowly utilitarian point of view says that we should be conserving it because of the direct benefits such as food, medicines, industrial products, that is tannins and everything that we get from it. Okay, so that is the reason. So you explain that properly. You should know the other reasons also. Okay, I told you broadly means you're talking about the ecological services, oxygen, aesthetic nature and everything out there. For example, you pay to get inside a place, right, where it's pretty. So that is ecological services. You're getting a benefit out of the ecological services which is already there. Now, mention the four evil quartet. We already mentioned about that. Give the examples for each of them. I spoke about that, but you should also be knowing. And among the vertebrates, which group of animals has the highest number in the global biodiversity? It is going to be animals. It has to be animals. So if at all you see this picture, Get to understand, it's always animals, but please know in invertebrates and this together it will be insects. Where in the case of plants, fungi has also been added. Should not be added, but yes, it is there. But anyway, the total number of species of fungi is the overall total of all these organisms out there except birds. All these organisms except birds. Okay. Now, alien species invasion caused decline or extinction of indigenous species. Justify the statement by giving 3 examples. Now, here comes the role of alien species invasion. I told you that you get questions related to that. So, Nile perch introduced into Lake Victoria in East Africa leads eventually to the extinction of ecologically unique assemblage of cyclid fishes out there. Okay, carrot grass, Parthenium, lantana and water hyacinth, they are also example. African catfish, Claris ganopanus, for aquaculture purposes is posing a threat to indigenous species in the animals of the fishes. Briefly explain the significance of David Tillman's long-term ecosystem experiments using outdoor plots. Yeah, this is something new. So, David Tillman said basically if there is more species diversity, the if there is more species diversity, basically the ecosystem is going to be facing less fluctuations or less variations out there. Okay, that is what it was said by David Tillman. And what is the use of it? That is what is going to be discussed right now. So, David Tillman long-term experiments conducted using outdoor plots are the significant in understanding the role of biodiversity in ecosystem. Increased biodiversity enhances productivity. Why did he say that there is lesser fluctuation if there is more diversity? Because, my dear children, if there is more diversity, basically there is more productivity as well. If plants are more, they are going to produce more organic matter. Other organisms can be stable because these organic matter are being produced by the plants. And the next thing, stability in the ecosystem is also increased. Why? Because over here, suppose a tiger is there, a tiger is there, rabbit is there, and there is another option. Basically, if the ecosystem just had rabbit, obviously, if the rabbit population goes down, tiger population also goes down. But you have multiple options, so basically you can sustain. You can still sustain with the help of that. So, diverse ecosystem makes more stability. Otherwise, what will happen? The tiger will be lost. It will be extinct. Resource utilization. Biodiverse plot utilizes nutrients and sunlight more effectively, minimizing wastage and ensure better ecosystem. So, if at all multiple organisms are there, obviously they will be partitioning the resources, having things, utilizing things even in a better manner. Okay, you won't be wasting off anything. Why? Because there are multiple organisms to feed on this. So, you do not need to waste on anything. You get it? So, that is one very important thing that we have. Cool? Yes. So, these were the experiments and species richness. Okay, this also I explained. So, this is just for you to understand that I have put over here. Okay, what is the other thing that I wanted to tell you from this particular chapter which is still important? Rivet-Popper hypothesis was given by Paul Ehrlich. He said that aeroplane is basically the ecosystem, and rivets, those bolts or screws which are there, they are the animals out there, organisms out there. So, if at all you remove it, the key species, obviously the ecosystem is not going to be stable. That is one very important thing. Apart from that, also remember there was an Earth Summit. Okay, there was an Earth Summit done in the year 1992. 1992. It was held in Rio de Janeiro, and it focused on basically biodiversity as well as sustainable development. Okay, they were talking about loss of biodiversity and sustainable development. This was the first thing. Then there was another summit, but this time it was World Summit. World Summit in the year 2002. So 1992 and 2002. This happened in Johannesburg. Here they spoke more about sustainable development. Rather than biodiversity. So both of them was there, but somehow the question is being framed. So please know, Earth Summit 1992 Rio de Janeiro, and over here 2002 you have the World Summit done in Johannesburg. Please know that. And one very important thing, I do not know how I missed it. Oh my god, just give me a second. So I'll write it somewhere here. Will it be okay? Yes, I'll write it here. So there are few species that got extinct, right? So I'll go a little top. Okay, over here when we spoke about IUCN list, I'll want to talk about list of some extinct species and the places where they lived. Okay, so first one is dodo. Dodo is a bird. It was found in Mauritius. Mauritius, please remember. Okay, second one that we have is quagga. Quagga. It was from Africa. Quagga, Africa. Then you had thylacine. It is from Australia. Then you have, they are all extinct organisms, okay? You need to remember their places also. Thylacine. And then what else do you have? You have stellar sea cow. Stellar sea cow, which got extincted because of overexploitation. Remember, it's from Russia. So please remember this particular list. This is super, super, super important. Okay? And apart from that, there were 3 species of tiger also. Java, Caspian, and I forgot the other name. Somehow forgot the other name, but please go through it. If it comes to my mind, I'll let you know. So these are the things which are very, 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 very important. Please know about that. And also one more thing, latitudinal gradient. It says that in the tropics there will be more, in the equator there will be more species diversity. When you go to the poles, it will be less. Okay. So in Colombia, which is more towards the equator, you will be having more organisms. In New York, it will be even more lesser. If you go to Greenland, it will be even more lesser. Okay, so Greenland is towards the poles. So these are few examples given in NCERT. It comes to my mind, and I'm just telling you that so that you don't miss out on anything. Apart from that, yeah, I think that sums up a lot of things. This should be more than enough. So I tried my level best to complete each and everything which is there in your NCERT. My dear children, this is what I taught. Please go through each and every question, and if you have time, just read your chapters as well. So if at all by any chance, if you have any other information as well which you want to read, you can go through it. Yes, I hope this video had helped you. I have put in a lot of efforts just for you guys. Please make sure you also reciprocate that. You also put the same amount, not same, multiple amount of efforts and do well. I'm praying for you and you're going to do good. Okay, wishing you all the best. Take care. Bye-bye.